All right. Looks like we're live here on Standing for Truth. I am with the man himself, the myth, the legend, Matt, or better known as Matt Man, Raw Matt. He goes by many names. So, Matt, how have you been, brother? Great to have you for this late night show where we are going to do a number of things. We're going to refute the chromosome 2 fusion, which has been completely overturned. It's been falsified. We're going to dismantle some of Gutsick Gibbon's video. I've already put out at least one article on the video itself and a 15 to 20 page comprehensive article essentially refuting everything there is to refute on the chromosome 2 fusion. So those two articles are linked in the description box, but we are going to tonight touch on just a few things. I'll play a couple clips from Erica's video proving that yet again we have an evolutionist with egg all over their face. And so this should be fun. We're going to comment on comments, Matt. And if we have time, depending on how we feel in an hour or so, we may open up the mic a little bit for some of our creationist brothers and sisters to join, give their input, ask some questions, maybe bring up some random topics like human evolution, the hominin fossil record, ERVs, whatever. We'll kind of go back and forth on it until Matt and I are basically ready to pass out. We've been doing a lot of work behind the scenes, a lot of the work mainly comprising writing. So again, I just put out a couple articles. I'm working on the Human Chromosome 2 Fusion Handbook. And so we've got a lot for people to look forward to, put it that way. But Matt, as you know, writing day and night tends to drain you. So why don't you, with that being said, why don't you kind of go over for a little bit? What have you been up to? It's a good time to announce you will be back in the debate octagon in June. You've got 2 debates in June. You'll be taking on Grayson, and you will also be taking on Mark Reed. So that's going to be fun. One debate is going to be on the age of the earth, and the other debate is going to be on evidence for evolution. So when I told the audience that this summer will be the summer of debates, I wasn't lying. We are going to have a lot of excellent debates for you guys to enjoy. Matt, over to you, brother. What have you been up to? How you been? What have you been working on?
I've been pretty busy, as you guys know, because I've been missing. So you probably haven't seen my content popping up very much. You haven't seen me very much. Mostly because I've been helping a friend that contacted me that has stage 3 breast cancer. So I've been going over to their house daily. Now their chemotherapy is completely over and they're recovering before they go into surgery. So I get a little bit of a break. And of course, a break for me is just more work with Young Earth Creation. So more writing, coming up with things, and of course, doing some debates. I figured I like to have some hard debates, right? I like the subjects that are really hard to defend. One of those things being extreme longevity, and the other one, the age of the earth, right? How can you defend a position where the evidence is against you? I'm like, bring it on. That's exactly what we do here. We attack the hardest arguments, and we give the audience the best answers that we can possibly give. So,
Amen. Well said. That's one of our goals here on Standing for Truth, is to provide everybody with the strongest possible model on creation that includes young earth creation, but also separate ancestry. And so, Matt, you and I are firm believers that the best way to answer the question of ancestry is in our genetics. It's our genes, traits, it's our genetics that are inherited, sperm and egg, not a rock, not geology, not geography, not a bone found in the dirt. And so we put a lot of work into the world of genetics and basically confirming separate ancestry, but also, as you said, providing the strongest rebuttals to the best so-called challenges and arguments coming from the evolutionist side. So whether that's chromosome 2 fusion, endogenous retroviruses, arguments against a literal Adam and Eve, arguments against genetic entropy, we are here to dismantle those. So Doki Doki Bible Club, appreciate it, brother. The articles that you definitely don't want to miss out on are right here in the live chat, but you'll also see them linked in the description box. So right here, refuting Gutsick Gibbon's video titled "Donny Botches the Human Chromosome 2 Again." We're going to touch on that a little bit. Bye-bye fusion is what I titled that one. Also, the overturning the chromosome 2 fusion. So basically, one article is specifically addressing Erica's challenge and then her follow-up video to that challenge. And then this article is basically just a comprehensive response in general to the human chromosome 2 fusion argument. I leave no stone unturned. Throughout this article, I put evolutionist claim. You know, this is the argument they're making. DDX11L2 gene's a pseudogene, something like that. And then I refute it comprehensively. So it would be a huge surprise, Matt, I think, to us both, if an evolutionist out there were to actually refute this article point by point, page by page, in a sophisticated and comprehensive manner. I really don't think it will happen, because the arguments that I refute in this article are basically the best arguments that people like Gutsick Gibbon have put forth in their videos. And just one by one, this article dismantles those. So, Matt, why don't we kind of just get right into it? We can start, unless there's anything you wanted to add, brother.
No, I do have some comments on comments to make later. I think that'll be fun twist for everybody. So, but we'll save that for later. We'll get into the nitty gritty, the technical stuff like usual. We can't help ourselves. So,
you know, we're going to be doing a lot of these too over the summer, just kind of impromptu response streams, impromptu comments on comments. Comments on comments are always fun. I know the audience enjoys those. We've done many of those in the past. And so because we get probably hundreds of comments a day, right, Matt, between both channels?
Oh, yeah.
Yeah. So we've got endless content there when it comes to comments on comments. Okay, I'm going to share screen here. I've got a dozen links open, so I want to make sure I get the right link. And then I am going to mute myself just so there's no echo over on my side. ZG says, why so late? Luckily, we do this full time. So we give you early shows. We give you, shows around 9 p.m. Occasionally, we'll get the 12 a.m. show. And sometimes we'll just get a show right in the middle of the night at 3 a.m. So we give you the best of all the worlds. Early, late, really late. Okay, so here we go. I'm going to mute myself, and we're going to start with this video over here, Gutsick Gibbon.
The comparisons that I'm running for Tompkins's chimp-human similarity are in like the downloadable version of BLAST with significantly more sort of coding involved. This is just doing it online in the browser, just playing around just to see how Donny's work here, his, I guess, recapitulation of Tompkins's work actually pans out. Now, my question is, what the hell was he talking about chromosome 9, about how you can find the fusion sites calling card in chromosome 9? Because as we just saw, you can't. You might be able to find like the telomere calling cards, in the telomeres of chromosome 9. But the only place that you find the fusion site in forward and reverse, it's in human chromosome 2. Nowhere else, as we just saw. So double check me, Donny. Go see what you can find. At the end of the day, this feels a lot like Donny just...
Okay, I almost want to play it again. So you heard her say it loud and clear. The chromosome 2 area, the so-called fusion site, the 798 base pair signature, that's the only place, she says. And she said this in her previous video where she initially made the challenge a year to a year and a half ago. And so she reiterated it there. I was going to play the first video, but she reiterated it here. So, and this is the most recent video from a few days ago. And so here it is, right from Erica's mouth. It's the only place in the entire genome, the chromosome 2, where you find the telomeric sequences, these repeats in both forwards and backwards, TTA, GGG, CCC, TAA, in a head-to-head fashion. And it's funny because the video she's commenting on And this is an issue, unfortunately, we get with our critics, is they're not actually engaging our argument. And so she was responding to this open mic debate, impromptu debate here that we had, where I engaged Mark Reed and Grayson for about 35 to 36 minutes. And so in this impromptu debate / discussion, I asked Mark, I said, do you hold to the same argument that Erica does, that basically the chromosome 2 area is the only place in the entire genome where you find these sequences in both forwards and backwards? And he says yes. Grayson, right off the bat, came out and basically admitted that, yeah, we do find these DNA elements in that specific order, in the head-to-head order and other chromosomes, because I've discussed it with him prior to this, since we had an entire debate on the topic. And so what I find funny is Erica, in her video, she tries to say that, oh, Donny, do you have the memory of a goldfish? And I'm wondering if she has the attention span of a goldfish, because it's impossible to miss in this debate. Firstly, she accuses me of just taking already published data from Dr. Tompkins and basically repeating it and just blindly trusting that it's true. Not that Dr. Tompkins' work isn't trustworthy, but my point is, this specific argument in this video that was laid out was a novel argument that Chris Roop and I uncovered by searching the genome using BLAST, searching the genome to see if what Erica is saying is true. That apparently there's only one spot in the genome that you find these sequences in a head-to-head fashion, TTA, GGG, CCC, TAA. And in this discussion that she's supposedly doing a response video to, she obviously rushed to it. She must have watched 3 minutes of it, got triggered, didn't even notice me saying this is data here in this document on the screen that Chris Roop and I came up with when we decided to test her argument. We wanted to see if that's actually true, because if that was true, that may be 1 point for the evolutionist side. Although there's 20 points against them, it'd be 20-1. They'd still lose, but at least they get 1 point there, right? That it's the only place apparently that we find these sequences. And so as you can see on screen here, it's impossible to miss me going through the chromosome 9 data. We also found many other chromosomes. You can find it in the article that I put out, that we find these sequences basically next to each other. Again, the sequences in forwards, the sequences in reverse. Admittedly, they're a little bit degenerate in the other chromosomes that we found them in, but here in chromosome 9, it's pristine. And that's why I'm emphasizing chromosome 9 specifically. And so if Erica actually would have watched the video and paid attention, she wouldn't have put out a video that was just so riddled with misrepresentations, misunderstandings, straw man arguments. And what's sad is I was looking forward to at least seeing her response in that video, but her whole video was based off it started off with her assuming that I was not answering her challenge about finding these repeats in forwards and backwards in mirror form elsewhere in the genome. And so her video is filled with rhetoric. It's filled with posturing. It's presented in an overconfident way, but it's all based on a false starting point. And so anything basically after the first minute where she introduces her video, first few minutes where she introduces her video, it just falls apart because she's arguing against something that I never said is the point. And so here's the video she's commenting on. You can go see it yourself. Here is the, you can go to the website, the article section. Here's the article itself that I wrote addressing her video. And so if you go down here, I put the visual of the chromosome 9 data. And so you'll notice, remember, and she even challenged me there to go do what I already did and present it in the video that she's responding to. I mean, it's unbelievable. You really can't make this stuff up. So again, she must have either just watched a few minutes of it, got triggered based on something that I said, assumed that I was arguing a point that I actually wasn't making, and then went and made this 30 to 40 minute video responding to something that was basically misrepresenting what I said in the video. So you'll notice here, chromosome 9, TTA, GGG, TTA, GGG, CCCTAA. So the point is, we find these telomeric repeats in both forwards and in reverse, the reverse complement, because DNA is double-stranded. We find it in the chromosome 9. So nobody's going to say that this has anything to do with a fusion. This answers her challenge. And this is something she did not address in her video. And her video, again, was responding to a video of mine where I actually presented this data, but it completely went over her head. So again, I want to show the audience, and then Matt, I'd like to get your comments of her issuing a challenge that has been answered. So here we go.
Taking Tompkins's word for things instead of running these tests himself, which again,
okay, we ran it ourselves. There's the data. So it's not relying on Dr. Tompkins. As a matter of fact, Dr. Tompkins, he did his own analyses, years ago or whenever it was, and he found these interstitial telomeric repeats, right, Matt, that are involved in gene expression. They're functional DNA elements important to the health of the cell. But he never, at least he didn't publish it. He did not find any instances where these sequences are in forwards and reverse. He never documented that. And when he was shown these findings, he was pretty amazed by it. And so the word is eventually he'll write something about it as well. But it was Chris Roop and I that discovered it. So it wasn't Dr. Tompkins. So again, this is just posturing coming from her. This is misunderstandings. This is all a big straw man. So let me play the next part real quick.
he has the tools to do this. I know he does, because like I've shown them on my videos, and he's gotten them from EvoGrad, so he could go and run this himself, but I guess he hasn't, because I don't know, he's not done it on camera, he's not up with. Again, this is just a web tool. The comparisons that I'm running for Tompkins's chimp human similarity are in like the downloadable version of BLAST with significantly more sort of coding involved. This is just doing it online in the browser, just playing around just to see how Donny's work here, his, I guess, recapitulation of Tompkins's work actually pans out. Now, my question is, what the hell was he talking about chromosome 9? About
Okay, so again, it's not a recapitulation of Tompkins' work, Matt. As you can tell, we went in, we tested her argument, we sought to refute her challenge. Is it actually true? Now, yes, she shifts the goalpost, she moves to the density. 50% of her argument's been destroyed. Then she moves to, well, the chromosome 2 area comprises the highest density of these repeats. Okay, well, we'll deal with that. I did deal with it in my article. But the point is, she has said over and over again, and evolutionists have repeated over and over again, that no, we don't find these sequences, TTAGGG, CCCTAA, okay, head to head anywhere else in the genome. And we falsified that argument. We absolutely demolished that challenge. And so they can't repeat that argument anymore. Let me continue.
how you can find this the fusion sites calling card in chromosome 9, because as we just saw, you can't. You might be able to find like the telomere calling card.
So what she means by this is what she's been saying, as you know, Matt, because we point out that there's nothing unique about the fusion site, because we find the so-called calling cards, the telomeric repeats, TTAGGG, in multiple places within the genome. You get hits everywhere on all the chromosomes, right? And there's been a lot of study into these sequences and what they do, at least one secular paper, but also some very sophisticated work done by Dr. Jeffrey Tompkins, demonstrating that these sequences are there for functional purposes, specifically in gene expression. Gene expression is important, okay? Gene regulation, gene expression, all of these different mechanisms going on in the genome with these DNA elements, and basically the ordering of them, these interstitial telomeric repeats being important. So she thinks that all you find is independent, isolated instances of TTAGGG here, maybe over here, CCCTAA as a reverse complement, but never head to head. And that's what she's saying. Well, maybe that's what they found. But as you can see, no, that's not just what we found.
Cards, you know, in the telomeres of chromosome 9, but the only place that you find the fusion site in forward and reverse.
Telomeres, Matt, you can go into this. Telomeres are the end caps of chromosomes. So yes, you find repeat after repeat, just TTA, GGG, CCC, TAA, and just the repeats. And these healthy telomeres come in, thousands and thousands, and yet the so-called fusion site's only 798 base pairs. It's weak, it's degenerate, it's very flimsy. But the point is, no, these sequences are found in the middle of chromosomes. These sequences are found scattered throughout chromosomes, and in chromosome 9, it's the same thing here. Okay, so what she's saying here is just totally false.
It's in human chromosome 2. Nowhere else, as we just saw. So double check me, Donnie. Go.
As we just saw, double check me. So again, everybody go to the article. Notice this, and I'll even go through this a little bit, okay? We also discovered a similar signature in chromosome 9. The telomeric-like hexamer sequence and its reverse complement are arranged head-to-head in a similar fashion to the purported fusion site. Was the reputed fusion site cherry-picked? It appears so. It is important to note that chromosome 2 is moreover the second largest chromosome in the human genome. Chromosome 9 is much smaller, and yet it contains fewer gaps between the telomeric repeats, with the chromosome 2 site containing numerous gaps. And so Erica wants to say, oh, but the chromosome 2 spot, it's unique in its composition. That's so arbitrary, that is so subjective, because we could say this area here in the chromosome 9, this composition is also unique. Take this signature, blast it out in the genome, and let's see exact hits on this composition. I'm not saying it doesn't exist, but let's see. Because again, there's things that are unique about the chromosome 9 signature here, okay, when compared to the chromosome 2. And there's things in the chromosome 2, like the high density nature of it, that are unique to the chromosome 2 when compared to the chromosome 9. Okay, one thing about the chromosome 9 that's unique doesn't make it a fusion. An example of an ancient fusion, in the same way that one thing unique about the chromosome 2 spot suddenly makes that an example of an ancient fusion. No, that's not how it works. Okay, chromosome 9 is much smaller and yet contains fewer gaps between the telomeric repeats. There's something unique about the chromosome 9, because again, we find in the chromosome 2 spot, it's incredibly flimsy. It is degenerate. Again, the intact ones that you actually find in the chromosome 2 spot, because the telomeric repeats that are actually found unbroken, there's a lack of those. But the intact ones that you find are typically found independently or in isolation. And I show that here. If you actually look at the, here's the cherry-picked fusion site. And what you'll notice is the actual unbroken sequences, let's say the TTAGG, they're found in isolation. You got one here, and then you just got a bunch of degeneration going on, basically, where it's not, these sequences aren't found in tandem repeat. And so when you actually look for the ones that are intact, you only find about 10 here with TTAGG, all found independently for the most part. Okay, here's one, TTAGGG, TTAGGG. When it comes to the reverse complement telomere sequences that are recovered intact, you only find about 43. So this entire region is highly degenerate. And when you actually look at the sequences outside of this region, okay, the sequences are very isolated and separated by hundreds to thousands of bases. So the entire signature just completely breaks down outside of this region. But again, healthy telomeres. Actually, Matt, if you wanted to cover this argument briefly, brother, healthy telomeres are thousands of bases long. And so if you had two healthy telomeres slammed together to form a fusion, we should find thousands and thousands of bases, but we only find 798. And so where they say this is high density compared to what? It's arbitrary. It's subjective, because if you're it's circular reasoning, they're assuming this is a fusion site. But if you assume this is a fusion site, then guess what? There's far too few sequences. It's too broken. It's too degenerate to be a fusion site. Now it's dense as a hotbed of genomic activity, basically, when compared to other sections like chromosome 9 and other chromosomes that have these same sequences. And again, the chromosome 2 is massive. I mean, it's the second biggest chromosome in the genome. Matt, can you touch on that a little bit? Just the fact that we find so few here, if 2 telomeres really did fuse, basically 2 chromosomes fused, end caps of the chromosomes, you'd find these sequences.
Yeah. Here's the thing about telomeres. There's 2 types. The reproductive ones are about 100,000 base pairs long, and the smaller ones are the normal telomeres that we find in somatic cells, and they range from 5,000 minimum up to 15,000. That's huge. So if there was an area that was more dense, or I guess what she means by that is packed with long telomere repeats that don't have gaps, because there's literally no such thing as densely packed telomeres. I don't even know what that means, because it's not a solid mass.
Well, that's a good question. So basically what she means by density is the fact that you have the TTAGG and then the CCTAA. You can see, this first bit, you got a bunch of TTAGGs. Your second bit of the 798 base pair region, you got a bunch of the CCCTAAs. And so by dense, she means you've got way more of these head-to-head arrays than you do in any other place in the genome, which is true. Because if you look at our chromosome 9 area, again, we find them meeting her challenge in forwards and reverse, head-to-head. But you'll notice there's not as many found as in the chromosome 2 spot. And so that's what she means.
I like how much more pristine it is, though, in chromosome 9. It literally touches. They go back to back with each other.
Right. And you actually get instances of, notice, TTAGGG, TTAGGG, TTAGGG. Look at, they're all just touching each other. They're unbroken. And then right away in the head-to-head fashion that she says doesn't exist, CCCTAA, CCCTAA. Okay, so over here, yes, you do have a lot of instances of the TTAGGG, but notice, they're mostly found in isolation. They're mostly found independently. The ones that you actually do find, okay? And you can highlight them yourself, and you're going to find that the intact TTAGGG sequences contained at the site, there's only about 10 of them that exist. There should be hundreds of them if this really was the ancient remnants of a fusion. And then you can highlight the reverse complement sequences, and you get CCCTAA. You get them, about 43 of them, I think it is. You get 43 recovered intact. And then outside of this region, the concentration of telomere sequences completely breaks down. Where you find telomere sequences basically separated by hundreds to thousands of bases. So that makes sense, brother. That's what she means by dense. This area comprises the most of these sequences, the highest density of them as compared to anywhere else in the genome. Anyways, good. Go ahead, brother. Continue.
Okay. Well, I'm with you, and I agree that there should be far more. And I don't mean you said hundreds more. I'm going to say thousands more. Remember, the smallest somatic cells that have telomeres have 5,000 minimum. So they need a lot more, and not just because of how many telomeres are base pairs long, and or that they could have been somewhat degraded, but the mere fact that there is a missing 10% of this chromosome in size. That's 25 million base pairs that are missing. Because think about it, telomeres prevent fusion. They prevent the head-to-head fusion event from taking place, because that's what telomeres do. Telomeres are your protective caps at the end of chromosomes that protect you from disease. They also prevent fusion from actually happening. So if they were massive in size, like she's trying to say that they were, like, oh well, it's so densely packed that helps their argument. It actually doesn't. So it actually helps refute it even more, because the more telomere sequences you find there, the more evidence would exist that it could have never happened. You get my drift? You know what I mean?
Absolutely. And so when they say, well, the reason why there's too few sequences, Matt, we'll have some fun here. I'll pretend to be the evolutionist. Well, the reason why, let's go to the fusion site. So again, I want to point out this area is, and I pointed out here, the telomeric-like hexamer sequence and its reverse complement are arranged head-to-head in a similar fashion to the purported fusion site. and it's a lot more pristine. Okay, it contains fewer gaps between the telomeric repeats, with the chromosome 2 site containing numerous gaps. Well, who cares? So the chromosome 9 spot is also unique in its composition, because that was Erica's rebuttal. She said, oh, so basically, she hasn't really acknowledged public yet, but we'll see if she does publicly, that yes, we found areas in the genome, specifically this one, where the sequences are found head to head. Okay. And she is going to say that, but the chromosome 2 area is it's its own unique composition. Well, so is the chromosome 9 is its own unique composition. Isn't that very subjective, Matt? Because we could look to all parts of the genome in many different ways, look at all kinds of DNA elements, DNA sequences like what we're looking at here, and find all kinds of unique compositions that are, let's say, unique to that area in the genome when compared to other spots. Isn't that right?
It's just typical evolutionary double talk. They would make great politicians.
Right. And so again, you'll notice there's only 10 you said they should find thousands of them, and then 43 of the reverse complement. Again, I don't know how she missed this. I guess I'd like to see basically her state in a video that, yeah, I didn't watch that whole open mic debate, because I said it many times. Mark Reed understood it. Grayson understood it. Everybody understood it, that it was Chris Roop and I who did this data. We came up with it, not Dr. Tompkins. From my understanding, he couldn't find this. Okay. So this is novel. And this is not recapitulating, as she said, somebody else's work. No, this is novel, and this falsifies her challenge. But the video she's commenting on, it's impossible to miss, because, I mean, even Grayson pointed out a couple times. He said, yeah, Chris Roop ran the blast. You guys did this together. So you can't miss it. So she says, I have the memory of a goldfish. She has the attention span of a goldfish, essentially. So yes, her challenge was met. It was demolished. I don't know how she missed it commenting on this video. And in the video, I present this, and you can see it on screen. I mean, it can't be any more clear. It's pristine.
She's not responding because she's got to go ask Dr. Dan what to say, what to do, just like she had.
How do I deal with this?
Has no clue, like she never had any clue with genetic entropy yet. She said multiple times in debates and conversations that she has no idea what she's talking about with genetics, yet Dr. John Sanford and Dr. Jeanson are wrong. And
well, she hasn't responded to this. Guys, I highly recommend check this one out. Christopher Roop and I co-authored this one, Refuting Arguments Against Genetic Entropy. Highly technical, comprehensive, and we absolutely dismantle the critics on genetic entropy, leaving no stone unturned. I mean, this is a very solid article that refutes these arguments, but you won't see them being touched or addressed by people like Erica or Dan.
Of course not. Remember, she's already thrown her hands up with genetic entropy. She said, it's nothing but error catastrophe. Therefore, I can just write it off and I don't have to answer it at all. She's already admitted defeat by just saying that she doesn't need to talk about it. Basically, it's like saying, you challenged me to fight, but you know what? I already had the fight in my dream last night, so there's no reason to do it. I already won.
Right. I won in my dream. Dodge, dip, dive, and dodge. And so you'll notice how these are unbroken. This is an unbroken chain of TTA, GGG, TTA, GGG. And again, to some people who don't understand this topic, or this is new to them, this might sound like a different language. That is just your repetitive sequence, essentially, for your telomere sequence in forward. This is it in reverse. And so you see them head to head. And again, unbroken. Notice this, CCC-TAA, CCC-TAA, CCC-T. And then it starts to break down. Go look at the worthless chromosome 2 fusion site. Matt, the first one you get is here. Several sequences in TTA, GGG. Look at this. All of this just unbroken or broken sequences. So notice how these occur primarily in massive gaps. You get what I'm saying, brother? I mean, these are occurring independently.
Yeah.
They're occurring in isolation.
Right.
You're telling me this right here is a result of a fusion when healthy telomeres, as you pointed out, represent thousands of sequences. But yet
Right.
we do not find that here. You should find hundreds to thousands of these. And then your CCC, TAA, the reverse complement of them, they also primarily occur in isolation,
They
independently
don't
in
even
big
function,
gaps.
you know? They see those sequences and they see them as dead, worthless junk DNA, and we talk about how they actually have functions. So they're reading a different book. They have a totally different view when they look at something than us.
So if they were to say, well, the reason why it's so flimsy, the reason why it's so pathetic, and I'd like to actually show the audience I did post the link. I know C. Brown might have wanted to join and give his thoughts on this, but if not, that's okay, brother. I understand it's late. So, okay. So I wanted to real quick, Matt, you're familiar with this. This is from one of the prominent papers on the fusion to begin with. And so I've got a lot of slides here, so I need to find it. And they point out, they ask the same question that we're asking, and then they follow up the question with really weak answers. So notice here from this secular paper, Matt, they're asking if the fusion occurred within the telomeric repeat arrays less than 6 million years ago, why are the arrays at the fusion site so degenerate? And so these guys, they like to posture and say, oh, this isn't an issue. This was expected. Oh, really? It doesn't sound like it was expected. It actually sounds like it was unexpected. They actually sound surprised. Isn't that right, Matt?
Absolutely. Because of course it would be shocking, because think about it. We have these sequences that are locked together. They're bound. Why in the world, if they would degenerate, would a gap fill in there? That would be just some random event. Think about that.
Right. And so if they say, well, Matt, the reason why it's such a weak signature, it's only 798 base pairs. It's, we only find a few of the TTA, GGG telomere sequences. We only find, about 43 of the CCC, TAA ones. It's because, firstly, when the fusion occurred, since telomeres are basically there to act as protecting end caps of the chromosomes, think of your shoelaces and you got that plastic piece at the end of it that basically, protects your shoelace from coming loose and flaring out. Well, what happens when that plastic piece breaks and degenerates over time? Your shoelaces start flaring out and they break down. And that's what happens with your chromosomes too over time, as your telomeres essentially break down and degenerate. That's primarily a reason why we age and die. Okay. And so they'll say, well, in order for this fusion to happen, since the telomeres are designed to protect chromosomes from fusing, the telomere, the end caps, they would have had to degenerate during the individual's life that the fusion occurred in. And then once it degenerated enough, the fusion could happen. And so basically the fusion started off as a weak signature. What are your thoughts, Matt?
So what they have to do then is say that sequence that they now see there is the one that existed during the fusion. They would have to, right? Because otherwise, they think about it. They have to say that these things broke down, but how? Okay, they broke down and then they immediately became attached. There would be no more wearing down at this point, right? So, like you said, if I wear down the ends of a shoelace and then I attach them and glue them together, and then what's going to happen? There's no more wear and tear in that area, right? It's only going to wear and tear at the other ends of those shoelaces now. So they're trying to say now, as a rescue device or as a retrodiction to try to make it force fit, is, well, maybe they wore down so much in the beginning that's what allowed them to fuse. Okay, well, they're far. What's the answer to the chromosome 9 then? Because that's not really degenerate, yet it looks like a perfect fusion occurred there. No one would ever say that one happened there. But then you have the chromosome 2, which has kind of just a messed up region where it doesn't really look like it was actually an active telomere to begin with. You know what I mean? Like, I could understand the telomere sequences themselves missing a couple of their sequences. That would show degenerate, not entire columns in between with entirely different code, right? It's not like all of a sudden you fused your two shoelaces together and all of a sudden there's multiple different brands of shoelace in there with it. That doesn't make any sense. But that's what we're seeing when we look at those sequences. They don't They don't have telomeric looking damage, which is in between those sequences. Telomere sequences are very easy to see. If we can go back to your chart too. Oh, go ahead. You got something to hold up.
No, you're doing good. I was going to say, what if they say, well, over time, that those areas were not preserved by natural selection. Maybe this whole area is not all that important. And so it degenerates over time, over 6 million years, from an original pristine state.
Well,
and so it
okay,
wasn't originally 798 base pairs with all these gaps and degeneration going on, but over time, mutations and the failure of natural selection to basically preserve this area has made it look the way it does.
Well, that would be easy because we have sequenced Neanderthal DNA. So why a lot of it may be degenerated, we would be able to see a more pristine fusion area if they were to run it there. So that would be easily checkable as well, because they believe that Neanderthal lived hundreds of thousands of years ago. So that to them is a very ancient human being that was more close to the fusion site than us. So therefore, that region should be much more littered with telomeric repeats than the weak degenerated version that they're telling us today.
Right, because basically, for the audience sake, is this specific type of DNA sequence, these telomeres that exist at the end of chromosomes, they're approximately 5,000 to 15,000 bases long. Okay, so think about this. If two telomeres fuse to each other, then you're looking at a signature or mark that's between 10,000 and 30,000 bases. But we only find, again, 798 bases. It's a very tiny signature. So Matt, if it's degenerating that fast, great point about the Neanderthals, right? So you're saying the Neanderthal, if we could sequence this area, essentially it should be a lot more pristine, since this would be hundreds of thousands of years ago than today, right? If you were to compare this site in extant humans and Neanderthals.
Exactly. And the same could be done with Denisovan if we actually have any of their DNA as far as that chromosome goes. I would imagine we found quite a few, but think about it. We have Neanderthal fossils that supposedly go back 430,000 years, right? So that's a long time ago. Therefore, some of their genetics should show a much more pristine fusion region with much more telomeric repeats that, according to them, would have degenerated in modern humans 430,000 years later. Just makes sense.
Right.
That's something they could run to prove us wrong.
Well, if it's degenerating that fast, right, because it's at 798 base pairs now, it's a very degenerated signature. If they really want us to believe that that's the reason why, okay, so what happens in another couple 100,000 years according to the evolutionary model? What happens in a million years? Is this signature just going to be completely wiped out, non-existent? I mean, if it's that rapidly mutating,
right? It would probably have to, right? I mean, if it's been this amount of time and there's those just so few left, especially on the top, right, and not the blue on the bottom. If there's so few of the red, then there's not going to be much evidence left of all. Look at that. Look at the red. TTA.
Right. So how long? So we've got these massive gaps. The telomeric repeats in forwards are almost non-existent. I mean, again, you've got what, 10 with only this area that's touching TTA, GGG, TTA, GGG. Look at the chromosome 9 spot that we found.
Much more pristine. So
much more pristine. Well, look at all next. Look at this. TTA, GGG, TTA, G. So notice how this is unbroken. No gaps for the most part. Over here, mostly broken. Yes. Higher density. Okay. So what? What is it? It doesn't mean anything. This is more pristine. So maybe the fact that this is more pristine makes it suddenly an ancient fusion, because that's a unique composition. You see what I'm saying? Why this whole density argument is just so arbitrary. It's so weak. It's so soft. It's so subjective. And most of the evolutionist arguments are just that. Very weak and soft.
Oh, of course, it's always a rescue device. It's always falling back on something else. It's always like, yeah, well, we see a fast mutation rate, but it must have been slower in the past, right? Why? Because deep time. The evidence goes against them. It's automatically the fault of something else. Always a rescue device. That's why they've been falsified so many times.
So do you find, so again, we have the authors of this paper. This is one of the prominent papers on the fusion. They're asking the question that we ask, that the evolutionists scoff at and say, oh, this isn't a big surprise. Well, you know what? It was a surprise to the authors that are at least somewhat looking at this objectively. Their conclusion's wrong, but they're asking what we're asking. Why are the arrays at the fusion site so degenerate? And that's a great question. And this question alone, how come they didn't say, the arrays at the fusion site are degenerate, Matt, and this is what we expected? How come they didn't say that?
Because they knew it was a shock. Just like they knew it was a shock when they discovered humans should have been far more diverse than they were. They'd use the same wording. Why aren't people more genetically diverse from one another? They knew it was a surprise. They knew it was a shock. They knew they didn't expect it. Why? What happened when they found junk DNA wasn't junk DNA? They said the exact same thing yet again. They always do that. They go, how it's a surprise that not more of this is junk.
Right.
It's just always a surprise with evolution, because they can't look outside the box. There is no alternative for them. It has to be one way or no way at all.
So that's why I find it very circular to say, but the so-called fusion site, the reputed fusion site, it's of a higher density. Well, by saying that, what do you mean higher density? What are you comparing it to? What are you assuming this is? They're assuming, right, Matt, it's a fusion site. And so They're saying as a fusion site, this has a high density versus other spots in the genome like chromosome 9, which they'll say is not representative of a fusion site. But wait a minute. If these sequences have been studied, they've been looked at, and in the genome, we've discovered that there's significant activity and function associated with them, then why isn't the fusion site just a higher dense region of functional activity in the genome? I mean, we understand, Matt, that the fusion site region comprises what? It comprises multiple start points for transcription, where RNA polymerase II binds. Okay, it's an amazing source of genomic activity. We've demonstrated that over and over again. And the fusion site itself, which isn't a fusion site as we're proving here, is essentially this functional component inside an essential gene, the DDX11L2 gene that's completely on fire and highly expressed. Matt, what do you say to the evolutionists that argue, oh, well, the DDX11L2, it's a pseudo gene. The activity there is just spurious. It's not really functional. You guys are just making that up. It's a good response.
I would have to say, okay, well, first of all, where is it expressed and how is it expressed? You'll be able to find multiple regions in the genome where it's completely expressed in high amounts, actually. It's just expressed very different, because we also find that same gene in other things, and it's expressed very differently. So I thought I would have had it in my notes over here, where it is actually expressed and how much. Very strange, I don't. I'm usually pretty good with my notes. Apparently, I haven't done this for a while, so
yeah, you're doing good, brother. I pulled up so people can look at this paper. We looked at this ourselves. We looked at it with C. Brown as well, who helped us, because again, the critics, Dr. Jeffrey Tompkins, he's done a fantastic job. He's done a sophisticated analysis of the DDX11L2, but the evolutionists hate creationists, right? The second they hear, oh, creationists did this research, it's like now everything else is just going to be in one ear and out the other. So we looked at it ourselves, and notice this, just look at the gene, right, Matt? It's right up on screen. I'll put it a full screen for people from current slide. Let's click hide. Look it up, the gene, DDX11L2, mayanlab.cloud. DDX11L2 has how many functional associations, Matt?
Okay, sir. 1,996.
Right.
Incredible.
Biological entities spanning 7 categories. You got all these different cell types, tissue types, gene types, and this is all extracted from 20 data sets. We've looked, you go onto this website and you got a drop down box basically for each functional association, each cell type. You can see what it's expressed in, you can see what it's doing, you can see its activity. I mean, this is not just a pseudo gene that's representing spurious transcription or just noise. No, this is pseudoscience. Evolutionary theory is pseudoscience. It hinders science. This is a highly essential gene. It's doing a lot. Okay, the gene functions in over 250 different cell types. What more do you want? You see, the evolutionists are some of the most biased, unobjective people in the world. Isn't that right, Matt?
Oh, they've hated us for a long time. That's why Erica's audience doesn't care what she says. As long as she says anything, they don't care. As long as it's against young Earth creationists, they don't care.
Right. So, is it valid in your opinion to say that what we're looking at here with the DDX11L2 is non-functional, represents
No,
junk DNA, spurious activity?
no, because a matter of fact, here's a good one for people. Look at what these things do, and then look at what other biological functions do that same type of activity. And DDX1L2 is doing a lot of different things, lots more than what they would even consider basic transcription factor of junk DNA. It's a step above that. So, you know, kind of going against what they claim as well. We also have a good chart that I found. Maybe I can pull it up, but I found it for you. It's on the human Homo sapien genetic database of the helicase 2, and it shows you at the bottom where those associations are and how much in the heart, the gallbladder, the body fat, the spleen, the testes, where it's actually a lot of it is concentrated. And it's just amazing how much function that they have in these regions, which means it's vitally important. If you find a lot of function in the testes, that means that it's obviously inherited. It's passed on. It's de novo, right? That's how important it is. It's not something even like an orphan gene, something it arises later when it's epigenetically needed. If you did a knockout test of this in your body, it would cause problems. Therefore, it's a little bit more than junk DNA. It's pretty basic.
So again, the arguments we're putting forth, right? The telomeric repeats, TTA, GGG, CCC, TAA, found in a head-to-head array at the chromosome 9, done in a novel discovery. This, the functional associations with the DDX11L2 gene, just go check the genetic database yourself and verify it. Okay. The DDX11L2 gene is also situated, as you know, Matt, in a gene-rich area. It's a region of the genome that comprises many other significant and highly functional genes. And it's ridiculous to say that this is simply genetic noise when this region comprises multiple start points for transcription.
It's what they do. They literally complain when they find something like that. Remember when they did the same thing with the Triceratops horn? They go, oh, who cares? It was in a Triceratops graveyard. He clearly pulled up a bull horn.
Yeah, right.
Oh, okay. So the fact that this is surrounded by other active genes doesn't matter at all. It's just arbitrary. Oh, okay. Typical.
Well, that's how unsophisticated these guys are. You know, I debated impromptu debate Dan Stern Cardinal. I mean, he couldn't get out of there fast enough. He knew he couldn't spar in the debate dojo on this topic, unfortunately, and his arguments were bad. I mean, he was assuming just because it's labeled pseudogene means that it is now essentially a non-coding gene that was once a protein coding gene that's broken and therefore doesn't have a function, and whatever activities there is spurious. He tries to say that the heterochromatin, these densely packed pieces of DNA basically comprising repetitive sequences, that's non-functional, and so that's going to be hit with mutations over time, and that's why apparently humans lack this ape-specific satellite. I mean, it's all based on a starting point that the majority of the genome is what? It's junk. But no, we now know in 2023, as we've advanced in genomics research, that genes are associated in very sophisticated networks, as you can see on the slide. And that is exactly why when determining function for a gene, let's say the DDX11L2 gene, you have to look for co-expression of these non-coding RNA genes. And there's, as a matter of fact, Matt, over twice as many long non-coding RNAs in the genome than protein coding genes. So yeah, you've got DNA transcribed into RNA, translated into protein, transcription, translation. But guess what? Just because something isn't translated into a protein, it doesn't mean that it's not doing anything or non-functional. We have a lot of functional RNAs that are basically doing the behind the scenes work, right? Matt, we like to use the analogy where you've got a movie where you've got the actors and actresses on screen and you're watching the movie play out, but what's going on behind the scenes? So much. I mean, your end credit scenes after each movie, I mean, it goes on for 5 minutes, it seems, because you've got people doing sound design, CGI, writing, producing, directing. And that's what we have in the genome is we basically, and that's why these protein coding genes tend to be more similar across creatures, right, Matt? Between, let's say, humans, chimpanzees, and other primates. But the non-coding RNAs tend to be less similar. Okay. Because they're doing a lot of the behind the scenes work for the respective organisms. Did you want to speak to that a little bit, brother?
Yeah, that's why we find them more taxonomically involved with things that are specific. And the protein coding genes are just more cherry picked because they're the ones that they can line up to prove their common ancestry. They don't like to compare them to the other regions of the genome. Matter of fact, they don't even like to compare some of the protein coding regions to each other. They don't like to compare cytochrome C to cytochrome B because that also builds a different tree. So they have to just focus on one specific area to build a tree that they're specifically looking for and ignoring the data in all of the other regions. For example, I always like to point out that if you were to do a paternity test, what would you do? You would look at the Y chromosome, the mitochondria, or the autosomal DNA. That's it, because nothing else can prove ancestry. What do? They ignore all of those and they only focus on a single protein coding gene. Cherry pick data to get the information and the data they need. Why? Because all of the observable evidence doesn't give them the answers that they want. Taxonomic boundary paradox? Oh, forget it. We'll just build our own phylogenetic cladistic chart. Why? Because everything else doesn't agree with us on evolution. So we have to build our own imaginary system. Go ahead. It's just imaginary. Easy for us to take down. Go ahead.
Hope, dream, and imagine that you, Matt, as a human, are related to a banana plant. That's science, Matt. Matt, you don't believe you're related to strawberries and banana plants?
Well, I'm glad the law doesn't, because I would be a cannibal. I'd be a cannibal if I'm eating those.
Right. With all the fruit that you eat, those coconuts and that coconut water that we basically, IV right into our bloodstream. I mean, there you go. Right. You're related to that coconut.
Yeah. In the imagination of only. We're still.
They hope, dream, and imagine. It's pseudoscience. This is a confirmed prediction. I want the audience to. Am I still screen shared? Let me make sure. Okay, yeah. Good. I want the audience to understand that this right here is praise I am, my brother. Junk DNA. Exactly. Junk DNA is junk science. Evolution, pond scum to people evolution, that is junk science. And so, Matt, we've done many predictions. We've had many novel predictions that have been confirmed, and that's what this is. Again, do I got to play Erica's video again? I mean, she literally says, no, what is Donny talking about? Chromosome 9. Sure, you find the TTAGGG maybe in an independent location, but no, the chromosome 2 is the only place in the genome that you find them head to head. Well, here you go. And we actually said, we need to look this up, see if this is true, because I mean, evolutionists, how many times do they make claims that you look into and the claim is it's false. It's just wrong. And so we basically made the point that we'd be surprised if that actually was the case. We find these internal telomeric sequences all throughout the genome, not just at the ends of chromosomes, but actually acting in gene expression. Okay. These are functional DNA elements important to the health of the cell. And so we predicted, you know what? If we search the genome, we're bound to find some instances of these arrays that Erica says doesn't exist. And so we went in, Chris Roop specifically did the blast analysis. And what did we find? We found that which the evolutionists said we wouldn't find. That's a confirmed prediction. That's science. Matt, what's the gold standard of science? Making testable and falsifiable predictions. And so if we say, you know what, This 798 base pair, this flimsy region that's supposedly representative of an ancient fusion, we're saying that no, it's not. We have things called promoters. They act as genetic switches basically in the genome to initiate transcription. Okay. And it turns out that there's 2 promoters in this site, and the second promoter is the promoter for the DDX11L2 gene that overlaps it. How do you get highly essential genes? By slamming together 2 chromosomes. And so we went in and said, you know what? If what we're saying is true, then we are going to find other instances of the TTA, GGG, CCC, TAA head to head. And what did we find? Without knowing, what did we find? We found other instances. That's the gold standard of science. Isn't that right, Matt?
Yeah, that's it. That's what we're known for on the Young Earth Creationists, the predictions that we make, right? So they're so much superior than the evolutionary side, and we just need the public to understand that. You see, the public doesn't even know that we have a model, let alone make predictions, and make these predictions are far more accurate than the evolutionist. That's what's astounding, and that's what the public needs to understand.
Well, and that's going to bring me to a clip coming up that I got, another one from Erica that I want you to speak to. You're going to get a kick out of it because, again, it's based on assuming that the majority of the genome is junk, including these non-conserved regions of the genome. Before I get to that, you're going to get a kick out of it, Matt. Notice this whole claim that it's spurious. Let's have a look. Is it spurious? Is it just noise? The fusion site sequence binds to at least 12 different transcription factors, including RNA polymerase II, which is the key enzyme that transcribes genes. Additional data in this paper will also show that along with the binding of RNA polymerase is the fact that transcription has also been shown to initiate, notice this, inside the so-called fusion-like sequence in a classic promoter-like fashion. Okay, transcriptionally active histone marks and open active chromatin profiles that are hallmarks of promoters. Notice this, the DDX11L2 long non-coding RNA gene containing the alleged fusion sequence encodes several transcript variants expressed in at least, isn't this what we said earlier? In at least 255 different cell or tissue types. The fusion site itself appears to be the functional start site for the shorter transcript variants. I pointed out earlier that the fusion site region, the 798 base pair region, comprises multiple start points for transcription where the RNA polymerase II actually binds. It's an amazing source for genomic activity. Anyways, I could go through these all day, but I also wanted to point out this over here, is you can do a blast analysis and you can find where these telomeric repeats, right, Matt? These internal telomeric sequences, you can find where in the genome they're located. And so notice we find them obvious. Okay, here's chromosome 2, second largest chromosome. You got your color codes here. But you also find what? You find plenty on 22, right? Look at all these on, and that's a tiny chromosome. What are they doing there? They're all over the place. Not just at the ends of chromosomes like Erica's saying. That's bizarre. 21. And I should say to people, I offered to debate Erica on this topic about a month ago, maybe. And for whatever reason, she didn't want to. So, you know, this is sound data. I'll probably do an open mic on this topic coming up and see if any evolutionists come in and take up the challenge. Notice 16. Now they're going to say, oh, but most of those represent just, isolated instances of either TTA, GGG or CCC, TAA, right, Matt?
Right.
But no, multiple of these, after we looked, actually represent instances of that, yes. But the fusion site too has many gaps, but also instances of a head-to-head array, like in chromosome 9 here. Here's chromosome 9. But Matt, what do you notice? Let me go full screen here. What do you notice, brother, about the chimpanzee genome and their internal telomeric repeats as compared to the human genome?
Kind of small, but it looks like how few
right
repeats there are.
Notice there's very few in the chimpanzee genome as compared to the human genome. So when they try and see, remember this quote: the differences make all the difference. So they want to look to things like synteny and homology and just shared sequences and similarities in gene order across chromosomes and so on and so forth. But they don't want to focus on the differences. Isn't that right, Matt? These internal telomeric sequences alone, same thing goes with the alphoid repeats that represent centromeres. They are found in abundance in the human genome, and you barely find any in the chimpanzee genome. Where's those similarities? How did that happen? What happened there? Did basically after the split in the human line, did humans just evolve all of these functional DNA elements and chimpanzees didn't? Or did the chimpanzee line lose them and the humans kept them? I mean, what story are they going to invoke there, Matt?
I know why. Like, why in the world would be the human have all these extra paddings and buffers? Because these telomeric repeats act very similar inside the chromosomes as they would at the very ends, where they're padding and protecting the chromosome itself from damage. Well, they're also protecting the inside from damage as well. So when you find them throughout, that's extra padding. Why in the world would a primate not require that? They're in the much more dangerous environment than human, which we change our environment. So if they're living in the jungle and they're going down and they're living in harsh conditions, clearly they needed a lot more padding. They have much higher methylation levels. So why not have even more buffer and padding? Because the rest of their genome shows that they have much stronger immunity to everything than humans do as well. So why would they not have more telomere repeats also as a padding?
Right.
Think about telomere sequences in general. Different animals have different telomere sequences, and we live a lot longer than chimpanzees. Did you know that? They have different length telomeres at the end of their chromosomes. Chimpanzees, gorillas, their telomere length is different than ours.
Is chimpanzee average lifespan something like 60?
60 max, yes.
Yeah. Okay.
We can double theirs, yeah.
So I want to ask you this question because you have such an amazing range of knowledge on it. The differences make all the differences. They don't want to focus on the differences. We point out the Y chromosome dissimilarity, which the evolutionists have failed miserably on that one. We've pointed to structures too, like the baculum, differences in the hyoid bone for speech, FOXP2 gene, but there's many other differences too. Can you briefly touch on those differences and really point out why these differences demonstrate the uniqueness of humans compared to chimpanzees, and why these differences would be very problematic for the evolutionary community to explain in terms of evolution?
Oh, yes. Yes, I could. I'm going to share my screen.
Sure.
Present screen share. Let's see. I am looking for...
And to the audience, if you do have questions, I understand this is a technical topic. It's an important one. Feel free to either ask the question in the live chat or even ask the question by joining. So it's up to you. I'll put the StreamYard link. Matt, take care of business, brother, and I'll be listening. I'll be right back.
Okay. I chose the wrong one. Oh, no. I chose the wrong screen share. Maybe I did. Let's see if this helps. Okay, everyone, I shared the wrong one. Maybe I can zoom out. Yeah, it doesn't allow me to change tabs. So when I click on another tab, you can't see it. Okay, so we're going to get into that in just a minute. I found this comment, which we were going to probably get into later. I found it hilarious. Somebody left a comment on my channel saying, Erica easily debated circle around you guys. It's not even remotely close. Johnny tries to continue to grift. So I said, how logical is that? Think about it for just 1 second. We actually post our debates that we've had with Erica on our channels for everyone to see. And she does not post them because she does not want anyone to see what really happened. Yet she ran circles around us. I think he's looking through the wrong lens of the camera. I mean, it's pretty logical, right? Why in the world would we post something that makes us look horrible, especially like that? It just makes no sense.
And okay, nobody hosts more debates than us on all sorts of topics. We've now hosted over 310. Nobody does more open mics than us. Every single month now, we've been doing basically a 6 to 8 hour open mic challenge to all evolutionists. And about a week, Praise and I, and Praise I Am, if he's in the chat, he can verify this. We literally send the link out to all evolutionists you can think of. PZ Myers, he refuses to join. Aaron Raw, Dan Stern Cardinal, Gutsick Gibbon. Where was Erica during that open mic where I was going back and forth with Mark Reed and Grayson? I know she was hosting another program, from my understanding, on the line, but again, we sent that out a week before and we went about 8 hours. So, we're here to defend our arguments, defend our model. We're putting on the open mics. We're putting on the debates. And Matt, we actually connected it was my second debate, but it was a debate specifically on ancestry with Erica. So I would say it's probably the most important debate that I've done with her on modern day debate. Your debate with her and John's, and we clipped them together for about 6 to 7 hours worth of debates.
Yeah.
If she was running circles around us, do you think we'd clip those together, put them out on YouTube for everybody to enjoy? No. The evolutionists, even your most informed, credit where credit is due. Gutsick Gibbon is probably one of the most informed evolutionists. That's why all the evolutionists are now going to her and her videos. But again, her arguments are not, they're not standing up under scrutiny, like the chromosome 2 argument. Her responses to genetic entropy, they fail, orphan genes, Y chromosomal dissimilarity, whatever it is. Truth Defenders makes a great point. He says, I can't get any theistic evolutionists to come and debate on SFT. Yes. Last year alone, the Evolution Debate Challenge series, we did, I think, 65. We all engaged in at least one debate on our team, right? You did at least one with Jackson Rowe on Did Man Live to 900? I did several on endogenous retroviruses. Kent, he set a record with about 50. Professor David McQueen did a bunch, T-Rock. So there's a team of us that's always willing to debate the science. We're a ministry, but we're also a ministry that actually frequently steps into the ring, right? The debate dojo. And we've got the evolutionary community on the defense here. And so the point is, the theistic evolutionists like Uninspiring Compromiser or, Ken Miller, Joshua Swamidas, Joel Duff, these guys, they're not actually debating. They're not actually defending the science. People like an Uninspiring Compromiser, he'll never engage an informed creationists like us on the science for Adam and Eve and so on and so forth. And so your theistic evolutionists, they're compromising, but they have no justification for their compromise. Could you imagine compromising, Matt, but then having no justification for it? If we were theistic evolutionists, don't you think we would know as much as possible about evolution? Don't you think we would?
Yeah, right. Of course. Yeah, it makes sense. They would you think they would try to make a model out of it too, right? Some have tried. Sentinel Apologetics. He wrote one, remember? He basically just took the evolutionary model and slapped the days of creation under different epochs of time. So he tried, that's something. But don't just say, I don't know, when you get an answer, because Sentinel Apologetics doesn't even know the science either. So no wonder he's a theistic evolutionist. He doesn't even know what he believes. He doesn't even know what they believe.
Well, because somebody like Uninspiring Compromiser, he'll say, well, evolution can fit in the Bible and we can interpret Genesis this way. And I'm sitting here like, listen, okay, go with your compromised interpretation. Okay. I'll take my interpretation where Adam and Eve were literal. They were created separate from any other form of life. Let's see what the science supports. And you're going to quickly find that they're reinterpreting Genesis for no reason at all, because guess what? That which they say can fit into the Bible, evolution, actually has no scientific support. So we might as well just believe that the moon is made of green cheese and somehow incorporate that into the Bible. Let's do a debate, the type of debate that Inspiring Philosophy likes to do. Let's title it, Can the Theory That the Moon is Made of Green Cheese Fit in the Bible? Do you really want to debate that useless topic? And that's all he debates. Can evolution fit within the Bible?
Right.
Is evolution even supported by the science? No. Linkage blocks. Taxonomically restricted and essential genes, genetic entropy, right? Mutation accumulation, DNA function, the chromosome 2. Ken Miller said what, Matt? If there was no fusion, if we can't find evidence for a fusion, there was no common ancestry with the apes. Well, as we can see here, the human chromosome 2 fusion has been what? Falsified. So according to Ken Miller, there is no ancestry with the apes. So to people like Michael Jones, to people like Joshua Swamidas, no, we don't need to fit evolution in the Bible in the same way that we don't need to fit hollow earth into the Bible. So, okay, Matt, go ahead. Is this your screen shared at the bottom, or is that mine, brother?
No, I can't get it to screen share. So what I'm going to do is I'm going to screen share. Oh, wait, maybe I can find it. Oh, yeah. Okay. I had it on just overview. So I was kind of messed up earlier. Look how long it's been since I've done this, right?
We're gonna get you a pro again in no time.
All right. So I'll try this one.
Oh, yeah. There we go.
Okay. We're going to go through these real quick then. We're going to show you the differences. Hey, little man. We're going to show you the differences between humans and chimpanzees in a plethora of different ways. I usually try to start at the top of the body and work our way down, but instead it's just kind of going to be random on this one because I didn't plan for this. So we have the hyoid bone for speech, which is indicative to only human beings. There's the studies to show that. There'll be links to a lot of these different things. We have a different shaped barrel, chest. We have a different number of ribs in our rib cage than the chimpanzees or any primate does. We have a 3-arch system in our foot that is not found in the chimpanzee. We have a subcutaneous layer of fat that runs underneath of our skin that no primate has. So this is what keeps us warm. It's what buffers us. They have a coat of fur. We have the subcutaneous layer of fat. I have no idea how that could evolve quickly or slowly. No one does. We have goosebumps. That we have, it's a human-only function. They do have functions as well. They keep us warm. We have hair on our head, very different than primates do. And another very interesting thing about this is that our hair grows extremely long, and it's the exact opposite on our supposed closest cousins, which would be the shortest. And again, male pattern baldness occurs very common in humans, but not in great apes. We grew beards. Evolution said it was from being punched in the face. They do not. But some of them have some facial hair. So I guess they are being punched in the face too. Who would have thought? I've never seen them boxing, but now we know why. Body fur. Apparently, that's right, everyone. Fur, I said it. Apparently, we lost ours. Why? Why humans lost their fur and became naked? Here we go. Chimps have thick fur, whereas humans have fine villous hair. The study also counted sweat glands, and they also found that the humans stood out. Shocker, I know, right? We also have under the microscope that we can see human hairs are very, very different than primates. Imagine if a forensic investigator came in the house and they found animal fur. And they are a human hair, and they identified it as a chimpanzee. They're like, well, a chimp killed him. Wasn't even a human. You would just laugh. You'd be like, man, ridiculous. It's so easy, though. I don't know what's going on over here. I'll have to slow this guy down.
He's saying amen, amen. We're not related to strawberries after all.
Yeah. So the differences in hair is absolutely ridiculous. We've got melanin in our skin. Primates have none. You shave a chimpanzee, a gorilla, any type down, they're not going to have any melanin whatsoever. We do. We have to have different melanin content because of based on where we're going to live is going to be based on how much we're actually going to be exposed to, how much sun and vitamin D we get. This is why females will always be more pale than males in any demographic of the world that you go to. So if you go to Africa and they have black skin, well, guess what? The females will be a little bit lighter. Why? Because they need more vitamin D to produce it for milk. Chimpanzees have none of this. Vision, we see completely different as well. Our vision is completely different. We're better at seeing moving objects than they are. We can contrast color as well. We see different patterns of that as well. The whites of our eyes, also very different than a primate. Theirs is very rarely white in most species, and sorry, I can go put an end to that if you want.
No worries, brother. Whatever's convenient for you.
We have tears.
I have 3 kids, so I'm used to it.
Right. Chimpanzees will not. They'll scrunch up their face, but they can't produce tears. They can't cry whatsoever. Only human beings do that. The blood is entirely different. Did you know we match pig's blood much more than we do actually our closest cousin, our chimpanzee? If you did a xenotransfusion, which is a transfusion from a chimpanzee, it would kill us. but the cells from a pig have a better chance of sustaining because of the shape and the size. It's actually much more close to us than it would be. That's why Eugene, I forget his name, Eugene McCarthy stated that we must be a hybrid between chimpanzees and pigs because we have more similarities to pigs than we do chimpanzees. There's alternative splicing, which shows a massive difference between humans and chimpanzees. There is the what is this? This is the skin microbes, completely opposite. Why? Obviously, they're covered in fur. We're not. We have skin, so of course that's going to be different. Here's orphan genes, completely different, not just regulation of where they exist, but where they're concentrated and what they do. In the human beings, they're concentrated in the brain. In chimpanzees, orphan genes are functional mostly in their skin. And here's the difference between the two. Here's orphan genes of the skin and the tissue types. Look at how different those are. And remember, orphan genes are taxonomically restrictive, so they only exist differently in each species, which are indicative to kinds, which I know the atheist evolutionists hate the word. Not my problem.
What if, Matt, if I could throw you a curveball, what if, because oftentimes the evolutionists, they'll respond like Erica, they'll say, well, orphan genes are species specific. And so they'll say, you creationists believe in species within a kind, let's say the canid kind, right? Where certain species that we would say connect to a common ancestor on the ark, they have certain orphan genes, taxonomically restricted genes, that other species in that same kind don't have. And so what's a good response to that?
Easy, because we're dealing with function. Of course, we're going to expect a steering wheel to be inside of a car and a truck and not on a motorcycle. So if we've got looking over at a cat kind, why would we need an orphan gene to help a feline be able to break down pure muscle tissue than us, which is going to be doing the exact opposite regulation? So what are we like as human beings? Very similar to a chimpanzee would be. Well, we would never normally be living in tropics. We would be doing that. We'd be identifying fruit to eat. So therefore, we would be having a lot of the same dietary requirements and needs. So of course, those similarities, we're going to have genes that are going to be function doing a lot of similar things. So orphan genes are, of course, going to be modulated and similar to an extent, and then they're going to branch out and have a massive difference amount because they're doing different things. Like I said, orphan genes are concentrated in our brain and in different capacities of our body, different regions of the body. So when we see different orphan genes in chimpanzees, sure, they can be the same.
Well, Matt, if I could jump in, I might have missed it, but I meant, so yes, we would agree that humans and chimpanzees are not related, right? So those orphan genes that we have that chimpanzees don't have, that definitely makes sense. They're taxonomically restricted, especially in the brain gene expression. But they're saying, let's say we have the dog kind. Coyotes, wolves, domestic dogs, dingoes, let's say, go back to a common ancestor. They'll say that the orphan genes that exist are species specific. And so a wolf might have a set of orphan genes that the coyote doesn't have. But we're saying that the wolf and the coyote are related to a common ancestor. And so their argument is essentially we're being inconsistent. So what would be a good response to that if evolutionists use that on you?
Okay. Again, I was trying to make the argument that the orphan genes are like any other gene inside of our body. They're just very small and they're unique, and they're called orphan genes because of the specific functions that they do. And a lot of functions that we do match that of chimpanzees. So of course we're going to share some of the similarities. So we can't say that it's inconsistent because we share a lot of consistent things with them. Then there's also a lot of differences, and we do have more differences when we look at those orphan genes as well. Not only the differences, but the concentrations of the ones that we share are different. So when we looked at that picture that I showed right here, these are shared orphan genes that we have with them. But you notice the wave and the concentration difference of where in the tissue we have? Massive. So those are orphan genes that we share. These aren't the different ones that they don't have. These are shared, and look at the difference.
So basically differences in like the brain,
yes,
with humans as compared to the chimpanzee, right?
Right, exactly. Here's shared orphan genes. Ours are functioning entirely different. So then we have our own branch of ones that exist only in us. So when they say, well, the dog orphan gene and the wolf orphan gene, they're the same. Yes, and they're functioning the same. Ours are not. So it might look the same. It might have the same base pair, but the sequences are different because they're functioning different.
What if there's an orphan gene in, let's say, the wolf, but that orphan gene in the wolf is not in the coyote?
Genetic loss is what we expect over time in our model. That can be explained very easily by us because we see genetic entropy, we see loss, we see tons of chromosome fusion in animals in the wild. And the more of this loss that comes out, especially when species are breeding at a younger age, they lose orphan genes over time, right? That was one of the predictions that I made. The African people groups will have less orphan genes than the rest of the people groups in the world,
Right,
because they'll be losing them over time because of their lower genealogical life reproduction age. So
okay, yeah, and I think that's the perfect answer. So basically, you have, let's say, your domestic dogs, your wolves, your coyotes, and everything in between, let's say, that go back to a common wolf-like ancestor off the ark. Though those wolves come off the ark, they begin to vary and change and speciate. Different groups go to different parts of the globe, and so subsets of the original orphan genes are taken into each new group or new species, and at the same time, some of the orphan genes are lost in some of the lineages, basically, because you're getting shifts from heterozygosity to homozygosity. So in some lineages, you're saying orphan genes could be lost. And so today we could look at a wolf that has a certain subset of orphan genes that would have existed in the common ancestor, but the coyote lineage would have lost those over time for whatever reason.
Correct. Exactly.
Yeah,
as things speciate, they lose more of that genetic load and information that's in their genome.
So it's not inconsistent then with the evolutionists say.
Right, exactly. If anything, they should be seeing more. They should be seeing the arisal of these orphan genes with new species that arise, because that's what they're expecting, right? Humans are a new species, so therefore, of course, to them, we should have more different and new unique orphan genes. So therefore, orphan genes should be rising in the population, not decreasing. And we see gene loss over time and decrease.
So basically, the archetype for, say, dogs would have had all of the orphan genes that we see in wolves, coyotes, dingoes, domestic dogs, but over time various lineages have lost their certain orphan genes. Other lineages have kept it, but it's basically a loss and not a gain of orphan genes over time, which is what the evolutionists would need. They would need new orphan genes coming into existence through mutation, duplication, de novo gene synthesis, but that's not what we're seeing. Is that right, Matt?
Correct. Even in a laboratory, they've taken fruit flies and they've forced reproduction into the point where there's no more orphan genes in those fruit fly species, and they can't get them back. They do not arise again. So they've lost their orphan genes entirely in a laboratory. In nature, that would take a much longer period of time, of course, because it's forced reproduction at a younger age. So, but
yeah, because in some of these fruit fly populations, they've forced these orphan genes out of some lineages, and other lineages have kept them, right?
Correct.
So the evolutionists are inferring erroneously that some lineages are actually gaining orphan genes, when in fact, no, those are the original orphan genes. Other lineages are having their orphan genes pushed out of the equation.
Exactly. They're all losing them over time. Some of them lost them even faster until they perfected the study model. And basically it was taking male fruit flies and forcing reproduction in a fruit fly that was very young in age. And then they said, oh, look at that. Their next generation has fewer orphan genes being produced in their genome. Let's keep doing that. After about 4 or 5 generations, they were completely gone. So the other ones, yeah, they still had orphan genes, but they obviously didn't force the reproduction to lower them entirely out. But that would be the end result and the outcome, and why I was able to make the prediction that the same would be true for human beings, in that the Africans would have fewer orphan genes, because as Dr. Jensen predicted, that they have a shorter generational lifespan when they had reproductive children. Well, that would make sense, well, observationally, that Africans will have fewer orphan genes than the rest of the human population, and that would confirm his predictions of what he predicted in the African Khoisan people.
Amen. Well, so you nailed it, brother. You nailed it. So that's from our experience, that is the main objection or challenge to creationists who utilize the existence of orphan genes as evidence for separate ancestry. And as we can see here, it's another unsophisticated argument. Isn't that right, Matt?
Yeah, they have so many, and you notice how many differences we've gone through. We've looked at like just a handful of things, and they're so different, so vastly different. They don't talk about these. Remember, they're only talking about the similarities. Remember, they only classify us as apes for 5 reasons. You know what I mean? And they ignore everything else because it was just a taxonomic classification. I don't think when they created that and he threw us in the primate family to call us apes, did he realize people in the future were going to be dumb enough to think that they actually are related to the people that they go and visit, the monkeys they visit in the zoos. I really don't think they did, but that's what we've got in today's world. So I'm sure that he would be rolling over in his grave if he found that out.
Well, you know, by definition, every creature on this planet has to be more similar to another creature. And it turns out humans are most similar in morphology and anatomy to a chimpanzee. But what happens if we remove chimpanzees from the equation, and chimpanzees no longer existed? There'd still be a hierarchy. The evolutionists would just say, well, we're the closest to a bonobo. Then you remove bonobos. Well, humans are closer to a gorilla than they are to anything else. And so it's a very, it's another arbitrary argument. It's just based on simple taxonomy, the fact that we can classify things, the fact that every creature is going to look more similar to some creatures and less similar to others. I mean, remove all of your great apes. Humans are still going to be closest to some kind of, you know, monkey, old world monkey, new world monkey. And so regardless, a hierarchy will exist, right, Matt?
Always. It has to, because, you know, God created things based on function. So we were related to an anteater a long time ago, and then they discovered primates, and then it changed because we have a lot of similar like functions. So we answer it better though.
Amen. Amen, brother. Well, unless you wanted to keep going through that, I could show the next clip, because the clip is surprise on nested hierarchies.
Oh, that's funny. Oh yeah, go ahead, because my list is gigantic. I could scroll down. I have about 75 more slides on that topic. So yeah.
You know, I'm thinking one day, and the audience is enjoying this, 40 people, even at 5 a.m. So we were just expecting a few people, Matt, because we've been doing stuff all day, and we thought, you know what? Let's go live, mostly impromptu, keep it free flowing and organic, and let's refute, you know, show the egg on Erica's face, which we've done. Because yet again, another evolutionist claim or challenge that's been addressed, it's been falsified, shown to be wrong. I mean, evolutionists have egg on their face in the world of junk DNA, pseudogenes. I mean, back in the day, the number one line of evidence for evolution was what? Pseudogenes. Shared genetic mistakes between humans and chimpanzees. They assumed that those were broken protein coding genes. They were once protein coding genes. And now we understand, no, these pseudogenes are functional. Many of them are essential. They are necessary to sustain healthy life processes in the cell. And so evolutionists who claimed that the existence of pseudogenes were some of the best lines of evidence for evolution, again, they were making claims without fully understanding the DNA language. Okay, so my brother Kevin, evolution really is bankrupt. It really is. It really is. I'm here to tell you, Matt's here to tell you, you're not related to walking sticks, grasshoppers, and apples. Right, Matt?
It shouldn't even have to be said. That's the world we live in, though. People can't even define a woman anymore. So, of course, we live in a world where people think they're related to bananas that they're eating for breakfast. Why? How is that a surprise, you know?
Right. Oh, bowling balls and elephants are both affected by gravity. And so they must descend from a common planet ancestor, maybe Mars. Mars is affected by gravity too, right?
Yeah, that's where the octopus came from.
Right. Dogs and trees both have bark. So they must be related due to that commonality. So all jokes aside, let's play the next, this clip I told you you'll have fun with. So you haven't seen it yet, Matt. So this is kind of off the cuff, but so this is a surprise. Sit back, relax. You're going to have some fun with this one, just like we had with the other. So anybody just joining us, we are going on about an hour and 40 minutes. And so for the first hour and a half, roughly, we completely decimated chromosome 2 fusion. We answered Gutsick Gibbon's challenge, where she said that there's nowhere else in the genome that you find these telomeric repeats in forwards and in reverse in a head-to-head organization. And we showed why that is not the case. We found it in multiple chromosomes, including the chromosome 9. So I want to recommend people to check out our website here. Click the article section. You can go to our blog. And I wrote an article here in the blog refuting Gutsick Gibbon's video titled Donny Botches Human Chromosome 2 Again. Read through this. The blog is limited in several ways with pictures and certain analyses that you do, and you might want to put it on screen like on blast. And so I do recommend going to the article section, clicking download PDF. You can zoom in if you prefer the text to be bigger and go through it here. So in detail, the ball's in her court now. You know, we already know how she's going to respond, the composition and the density, but that's been refuted in this article. It's also been refuted during tonight's show for those who like a video rather than something to read. Also check this article, Overturning the Human Chromosome 2. This is comprehensive. This is basically a sneak preview. This is a snapshot of the book, Chromosome 2 Fusion Handbook, that I'm working on. And so this gets into basically everything that has to do with the chromosome 2 fusion. Erica put out a video, I don't know, a year and a half ago, where she tried to refute our arguments. She claimed that, you know, the DDX11L2 gene is a pseudogene. I refute that here. Ken Miller, he's made various arguments. I refute those here. Another claim they've made, creationists don't know what the fusion site is doing. I mean, what a ridiculous argument that is, but they say it. And so I refute that here. They look to a homology argument. I address that here. They also look to the argument from synteny, and I address that there. So take advantage of the material and the content that we are putting out for you guys. We want to get you the best and most up-to-date material on the topic. Okay, so that was just a recap. Al, appreciate it. Donny and Matt, the dynamic duo. Matt, remember when we did that? We were refuting a critic maybe a year and a half ago on speciation, post-flood speciation, and we had that Mattman call. Do you remember that?
Yeah. I'd play, hit the button, I'd run in.
Right. The show would start with just me, and then I'd see a really bad argument from the critics. And oh no, you know, this is a job for Mattman. And we put that classic Batman TV show with the POW and the BAM, Adam West, I think it was. So good times, brother. Okay, here we go. Surprise clip coming in, Matt.
All right. Take it off mute. I hear nothing.
Oh, I must have forgot to click share audio. Okay, let's try again. Entire screen, share audio. Okay, here we go.
Pretend for just a few moments to be a naturalistic evolutionist and lay out the strongest case that they make for their position so that we can create a steel man, and then you can put your feet back in the Christian shoes and refute the straw man.
Lyle starts off by doing an okay job with this. He goes on to go into the specifics of the arguments that would be made, and he starts with this one.
One of the arguments they would make is they would make an argument based on similarity, taxonomy, the fact that we can classify organisms, and they fall into a nested hierarchy. So we know a hierarchy, you know, this these two things belong to this, and this belong to this, and so on. And there's a hierarchy within a hierarchy, so it's a nested hierarchy.
See, this is good.
So you can classify humans. We're in the the kingdom, phylum, class, order, family, genus, species. So we're in the primates, and we're in the mammals, and we're in the vertebrates, and so on. And they would say that that's what we would expect if life evolved from a common organism. We'd expect to be able to classify it that way.
This is even better. At least we know that Lyle accepts that morphologically humans are primates, mammals, etc.
And then I think they would further try to argue. They would say that not only is it true in terms of physical traits, but genetically. Genetically, we're more similar to, say, a chimpanzee than a lizard, and more similar to a lizard than a fruit fly, for example. And so they would say that genetics, the genetic code, also falls into a nested hierarchy. And so they would say that then is support of common descent. So that's where they would take it in terms of the genetics, I think.
Yeah, absolutely. We should see the nested hierarchy both in morphology and in the genomes. We should see organisms nest respectively in larger and larger groups moving upwards, or more and more specific groups moving downward on taxonomic rank. You did leave one important thing out here, which is that organisms are nested in their genetics, yes, but specifically they are nested in both functional and non-functional regions. The fact that they're nested in the latter portion falsifies the idea that it's common design, not common descent. Because unless God is just being very tricky, I don't see any reason that's been put forward by intelligent design advocates for there to be a nested hierarchy in the functional regions, which would be in accordance with design, and the non-functional regions, which should not be correlated at all, because they don't do anything.
And they might.
Wow.
Hashtag weak and soft in terms of the argument, right? Another weak argument. Notice the built-in assumption. So the functional regions form hierarchies in the biological world, but it's the non-functional regions that form hierarchies in the biological world that falsifies the common design hypothesis. So she's acknowledging that the functional regions, or the function associated with physiology, morphology, anatomy, and genetics, those falling out into hierarchical patterns in a hierarchical distribution, yeah, that can be explained through common design. But the non-functional regions that fall out in a hierarchical distribution, that falsifies the common design model. What is she assuming? That these regions are non-functional. You see how they assume the genome is junk? They assume these highly conserved regions are functional, but the non-conserved regions are, they've been hit with a lot of mutations over time because they're not really that important. Or they'll look to, third position codon variation or neutral variation that falls out into hierarchical patterns. Herman Mays likes that as well, these statistical hierarchy arguments. And so again, the assumption is that we're looking at a lot of non-functionality. Anyways, what was your overall thoughts of that, Matt? Is that immediately what came to your mind as well?
Yeah, it's like they just impose what they think would falsify us based on their interpretation of the data, and then says, "See, it's why would God do that?" They do the same thing. They go, "Your God is trying to trick us all the time." I would be, and it's like, wait a minute, no one was tricked up until recently, and then this new mindset comes out, and now all of a sudden it's God's tricking us. No, your interpretation of the data is what's tricking you, and then you blame God for it tricking everybody. It's like, wait, you're the one that's believed the lies of Charles Lyell and Darwin, and it's just gone down and politically pushed, and the agenda hit the mainstream, and then the Rockefellers pushed it on the public education system because they're funding it, and then all of a sudden now that's true. Okay, all right, go ahead. You can believe that all you want and call it science, but don't pretend that that model has anything to do with ours, because they couldn't be more opposite. And we've already explained how we would expect these things to play out. I gave a presentation on it. I have a book on it, on how we explain the diversity of life and how it makes much more sense, and how they cherry pick data to even get those trees. Like we just talked about, they have 2 cytochrome B and C, which are conserved regions, right? Protein coding regions. They ignore one because it doesn't make the tree that they want. So they invoke multiple rescue devices for why that is. But no matter where in the genome you pick, it's going to build a different tree. It's just not the tree they want.
Incomplete lineage sorting, basically, depending on what gene or sequence of the genome you look at, you can get totally different trees. The Y chromosome dissimilarity, where's the hierarchy there? In the Y chromosome, humans, in overall architecture, size, and gene content, humans are closer to the gorilla than they are to the chimpanzee. Evolutionists say that the chimpanzees are closest cousin, and we go back to a common ancestor. But we were talking about earlier, a relative hierarchy is expected regardless. I mean, we're created with DNA, RNA, proteins. We're created to eat foods, digest foods. We are created with similarities to basically everything in the biological world. And so by definition, we're going to have to be more similar to some creatures, less similar to others. Turns out we're most similar to pick any animal on the planet, Matt, and it's going to be more similar to another creature. Pick a fish. Is a fish most similar to a human, or probably most similar to another type of fish? Are vertebrates overall more similar to other vertebrates, or more similar to invertebrates? No, I mean, it's ridiculous. Take away chimpanzees, take away bonobos. There's still a hierarchy, because the evolutionists are going to say humans are most similar to gorillas. Humans are more similar to gorillas than they are to a lemur. But the biggest thing, Matt, that I think we should address is this idea that because the non-functional regions also fall out into hierarchies, why should that be? She's assuming, and this goes to a lot of the non-coding DNA. Yes, they expected some function in the non-coding DNA, but not nearly as much as we've discovered. Okay. And so a lot of what they assume is non-functional and that falls out into hierarchies are what? Pseudogenes. They are endogenous retroviruses, allele sequences, at least with primates, and just non-coding regions of the genome. But we pointed out earlier that it's your protein coding regions of the genome, your protein coding genes, that tend to be most similar. And a lot of your non-coding DNA tends to be taxonomically restricted, because your protein coding genes represent a lot of times those major similarities in structure between us and, let's say, the chimpanzees. But notice this, the functions of non-coding DNA: regulating expression of genes, maintaining genome integrity. There's also a lot of built-in mutational buffer and redundancy. Okay, provides structural elements to chromosomes. A lot of the DNA there is not just junk or filler. It's actually there assisting in chromosomal architecture and integrity. Okay, if those fall out in hierarchical patterns, yeah, function. That's what we predict is function. Hierarchies by design because of function, rather than hierarchies because of descent. The hierarchies because of descent that Erica's pushing would predict a lot of non-functionality, evolutionary leftover, a lot of viral fossils in the genome, genomic mistakes. Okay, turns on and off genes. There's a lot of regulation going on in these non-coding areas of the genome. Provides origins of replication, works as regulatory elements during transcription and translation. I mean, so on and so forth. The pseudogenes that fall out into hierarchical patterns, same thing. We have function after function. And here's the thing. They're going to say, because we have evidence for genome-wide activity, roughly 80%, okay? Transcription-wise, we're probably looking between 60 to 80% of the genome is actively transcribed. Now, they want to say, right, Matt, Erica would say, well, most of that is noise. Most of that isn't really doing anything. But the problem is, and I pointed this out in my latest discussion with Dan Cardinale, I've pointed it out in several of my debates, transcription is an energy-intensive and resource-intensive process. Nucleotides are actually being utilized to produce RNA molecules. And if those just end up being noise or spurious, that's problematic, because that is draining energy for no reason at all. Since this process of transcription is an energy intensive process and the genome is using up just a large amount of energy, ATP, right, to produce these transcripts, then it makes sense that these transcripts are being produced for a reason, or else this would be wasteful of resources. And that waste should be minimized. And therefore, there shouldn't be as much activity in transcription, because that transcription should essentially be suppressed. Or all of these evolutionary leftovers should over time be removed or done away with through what? Through natural selection. And they'll say, oh, well, sometimes it's easier to just transcribe this junk and have a lot of noise rather than get rid of it. Oh, really? Clutter is better then, Matt? Clutter is better? Notice this. Just cluttering in general in your life. Clutter can drain all of your energy. Look, here's an example with people sometimes in their home. My car sometimes, especially when I was in college, it would get so cluttered, right? And it would just anxiety causing stress. And once you declutter it, I mean, it's such a breath of fresh air. Same thing in the genome. If the genome was mostly junk and a lot of this was noise, it would make sense that there would be mechanisms in the genome to declutter it. And that would help the genome be more efficient, not keep it around. I mean, I can't believe evolutionists literally say, well, it's easier to just keep it around. It's easier to just transcribe this noise. Notice that. Yeah, go ahead.
Did you catch that though? They immediately invoked that that selection wouldn't remove them. But what are they? The first thing they said to us when the mutation rate was too fast, that selection removed everything.
Yeah.
They did the exact opposite.
Selection is their god, right?
Selection. It can do anything. It can ignore things and it can choose things, but only if it's in favor of evolution, because it's got to be true no matter what.
I mean, look at everything that goes into just transcription. You know, you got these different phases where, right here, transcription ends in a process called termination. Termination depends on sequences in the RNA, which signal that the transcript is finished. Before transcription can take place, the DNA double helix must unwind near the gene that is getting transcribed. So you got initiation, elongation, transcription, termination, like we just talked about. Notice this, ATP. Matt, talk about ATP a little bit. It's needed for transcription. Again, this is an energy intensive process. Does it make sense that most of this activity is just noise and mostly spurious transcription?
Man, ATP is the energy of every cell in your body. It's required. ATP is the cellular energy warehouse of every living cell. So imagine the energy that's required to produce and run and utilize ATP going on and everything in the body. It's not wasting. Your body's smart. It's requiring you to use only what it can to survive. It doesn't waste voluntarily good energy in places that are useless. There's no reason for it to. Glucose is utilized very, very smart by the body. And that's why we store body fat so quickly, because our bodies are smart. They're like, hey, if there's ever a famine, we have got to have some reserves coming on because we don't know when that's going to be again. So it's using it for all that ATP and energy in the body. So just imagine the body wasting it on something that's been junk, not for 10 years, not for one generation, not for 1,000 generations, hundreds of thousands, millions of years. It makes no sense. None.
No, it really doesn't make any sense. And here's the thing. If transcription factors are just binding randomly over the genome and there's nothing important going on here, that would muck up. That would gum up the systems of the cell. And it would actually create what's called an interference problem, where the operations in the genome are being gummed up. And this would be a mess. This would create a mess. And so these are the types of pseudoscientific arguments that evolutionists look to. Okay, there's only so many copies, Matt, of a transcription factor that are going to be produced. And if these transcription factors were merely binding to random sites in the genome, there wouldn't be enough transcription factors available to actually do the binding that's necessary for these transcription factors to properly function. Okay, so there's numerous reasons why everything going on in the genome and everything that the ENCODE project has detected is for good reason, is for functional purposes. And therefore, and this is where I wanted to go to some nested hierarchical diagrams here. If the majority of genomic sequences and elements are there for functional reasons, then guess what? Because again, Erica is assuming, so here, humans nest within primates, primates nest within mammals, right? Humans and primates are all mammals. We're also all vertebrates. And so all of these nest within vertebrates. So humans, yeah, are more similar to those in the primate family than either are to a whale, and a whale's a mammal. So, okay. But again, if these hierarchical patterns are there for functional purposes, it's no surprise that a human is more similar to a dog. Notice here, It's no surprise that a human is more similar to a dog than a human is to a fish. Is that really surprising? And on a genomic level, everything that forms hierarchically, like mitochondrial DNA, right, Matt? Like these nuclear DNA genetic markers, it's for functional purposes. And so Erica is, if you could speak to this, she is assuming evolution to prove evolution.
She has already started out completely wrong by conflating that a tree can be made based on evolution. So for an audience that's listening, you're probably asking yourself, well, why can an evolutionary tree be built at all? The reason it can be built is because of young Earth creation. And I'll tell you why. Because in the 1960s, Ernest Mayr was an evolutionary theorist who predicted that if deep time really is true and we did descend from a common ancestor, so much time has passed that there should be no genetic similarity left in anything on Earth, because so much time has passed, it would have eradicated all genetic similarity. And you know what? He's true. He's right. But there's another twist to that. In the 1970s, along came the young Earth creationist, Dr. Henry Morse. And I think he might have even started Answers in Genesis, or one of the major Christian. Yeah, he created one of the major organizations, and he predicted that because the same common designer created all life, we would actually find great genetic similarity in all life on Earth. And because not a lot of time has passed, we will find a lot of genetic similarity in all of nature. Guess what happened? The prediction of the young Earth creationist came true. Evolution was falsified. Evolution swept that under the table and made a retrodiction saying, well, we invoke a global bottleneck must have happened probably 200,000 years ago that wiped out all land life and all aquatic life, reset the genetic similarity pattern that we see. And that's why we still see genetic similarity, because the global bottleneck, which is actually, again, in our model, not theirs. They invoke it. They looked in their fossil record. They never found it. It's ours. Yet again, they took from the young Earth creation model, collected it as theirs, ignored another falsification, and then went on and now use it as the best evidence for evolution, building their evolutionary trees when it's originally a falsification of it. So never let an evolutionist get away with that if it comes up into an argument.
It's a great point. I mean, look at this classification of automobiles. And then they'll say, oh, cars don't reproduce. Oh, thank you, evolutionists, for stating the obvious. So adding reproduction helps them. That adds more complexity, right? That adds more of a problem for the evolution model. Isn't that right, Matt?
Oh, of course it does. That's now you've got living biological material that you have to deal with and cope with. And I bet you anything, if we were to reverse this argument that they've been using on the chromosome 9, I bet you, if anything, if they had found chromosome 9 first, that they would be using the exact reverse argument now, saying, look at this. It's a clear head-to-head telomere, and if the pattern is so locked up and perfect, you're pointing at some remnant that looks like it's not even connected. The pieces are everywhere. Sure, it's more dense, but who cares? Look how degraded and messed up it is. That you can find that throughout the genome. That doesn't mean anything.
Exactly. You know, I won't be surprised if an evolutionist eventually says, you know what? The chromosome 9 may just represent an ancient fusion.
Yeah, it's probably the original. And that explains why it's missing 25 million base pairs on chromosome 2.
And, you know, they could say that the chromosome 9 actually represents an earlier fusion because it's more pristine. Or they could go the other way and say, well, it's not as dense, so it's an older fusion. So do you see how the evolutionists, they up, down, both evidence for evolution, blue, red, black, white, whatever. It's all evidence for evolution, right?
Yeah, always unfalsifiable. No matter what you do, no matter what they ask for, if you find it and you give them a better answer, it doesn't matter. Their money is in it. They've invested their time in all that school and education and the money, and their business now relies on that. They're not going to give this up. They don't care how much evidence there is. Their livelihood is invested in this. That's what the sunk cost fallacy is. And they're the epitome of the sunk cost fallacy.
Matt, what do you mean? Don't you see all the similarities with your grandpa here? I mean, look, you got binocular vision, nose, teeth, hair, very similar smile.
People believe it, man.
Evolution. There's your grandpa. That's science, Matt.
Yeah, the evolutionists, they see that, and I just feel like somebody made a monkey out of them.
Right. Yeah. Oh, because we both have binocular vision, so there's evidence for evolution. Notice this. Now, if evolution were true, and it actually turned out that all of this so-called junk that they have was removed, there's good evidence for that, they'd be like, see, here's evidence of selection working, or here's evidence of these mechanisms in the genome suppressing all this transcription. So basically they want us to believe, right, Matt, that pond scum evolved into people.
Yep.
That fish evolved into fishermen, that you had a single-celled like ancestor. Well, basically, no, you have non-living chemicals in some warm pond. So you have chemical evolution or abiogenesis. They somehow come together to create the first self-replicating molecule. Then you have, single cell to multi-cell evolution. And then you have, the evolution of a fish. And then the fish comes out of the water, Tiktaalik style, right? Fish, amphibian, reptile, mammal, eventually humans. And so through selection and mutations, epigenetics, whatever, everything going on, the environment interacting. But no, selection's just incapable of removing all this junk. So, like, do you see how sometimes they invoke basically magical powers for selection mutations? But in other instances like this, with junk DNA, it's so limited, it's weak. They want their cake and eat it too, right, Matt?
Of course they do. They see exactly what we young earth creationists talk about all the time. We see, when we look at a evolutionary tree, we see the ends of the branches, or what they call nodes, and we only see that. That's why Ken says dogs produce dogs, right? But what do they believe? They believe that there was a root of a tree, and then the trunk of a tree, and then the branches. And they agree, oh yeah, we're at the end of the branches. But guess what? Evolution is never supposed to have stopped. We should be seeing linear evolution existing today all around us, because that's what they predicted. And they said, oh, no, evolution can do anything, and you don't know what you're talking about. There's no way that the thing's supposed to be evolving in an upward progression. Oh, really? That's what your books show. That's what they tell us. So what they're admitting is that they don't see it, and it's not true, because we're only at the ends of the branches, which is exactly what the creation model would say, which is always said. So it's ironic.
Well, Matt, your fish to your fisherman, mutation, selection, lots of time. Or they say, they would admit a frog turning into a prince instantly because the princess kissed the frog. That's a fairy tale, right, Matt? But some kind of amphibian frog-like creature turning into a prince, a human, over millions of years of selection and mutation. That's science, Matt. And if you don't believe it, then you're just rejecting science.
They like to play the victim card and put you on the defense with nonsense like Bill Nye did with Ken Ham, and throw out ludicrous questions all the time. And they do it with everybody. And they don't realize how ridiculous their concepts are. And I think if you question them on it, you know how you know they're embarrassed about it? Call them an evolutionist. Watch how mad they get.
Right. Oh, that word doesn't exist, Matt.
That doesn't even exist. How many Christians get mad when you call them a creationist? None.
Right.
Are heads on tighter, or are they just like triggered snowflakes, or are they just that embarrassed about what they believe?
Or the ones, especially on Facebook, I'm in these Facebook groups, right? And 99.9% of them, these thousands of evolutionists, trolls, most of them don't even have real accounts. You click their Facebook profile, and it's like a picture of one of their favorite sci-fi characters and fake name. They literally just made an account to troll. And a bunch of coward evolutionists on Facebook refuse to debate. A lot of them say, evolution's consensus. So why would I debate consensus? Why would I debate evolution? That's fact. I'm not going to debate. But they'll spend their days and nights in a Facebook group trolling creationists. See, Matt, if I told you that Superman could fly quickly through the air, right? You'd say that's a fairy tale. That's comic book stuff. But if I told you Superman could fly slowly through the air. He can fly, but he's got to be slowly. Then that's science, isn't it?
That doesn't make it more real. I'm sorry. Yeah.
Exactly. It's
They're
both fairy tales.
they're arguing what color socks Santa wears. Guess what? Santa doesn't exist. I'm sorry. You time to grow up.
Time to grow up, evolutionists. Okay, the tooth fairy. I understood when you were a kid, you believed when a tooth fell out and you'd put it under the pillow, the tooth fairy would fly in and leave you $1. Okay, very cute. Then you learn that's a fairy tale. Okay, your favorite Disney movie is The Lion King. There's not really a lion that can talk named Simba. It's a fairy tale. And you know what? You're not related to banana plants, whales, and puppy dogs. That's a fairy. It's time to grow up, right, Matt?
It's just embarrassing. It's sad that adults believe this. When I'm standing in the store and I look over at this fat dude, at the Slurpee machine slamming down his drink, and I'm thinking to myself, man, I bet you he thinks he's an ape. I bet you he thinks he's related. I bet he does. I wear those creation clothing, right? And they just look at it, and you can just see him get mad. It's like.
Matt, but people wonder how we have so much energy, how we can do so much work, put on so many shows. If you can see in the camera, evolutionist tears.
That's why
you start your morning off with a nice fresh cup of evolutionist tears. You're not even gonna have to sleep. Let me there you go. So anybody, if they're wondering how to have unlimited energy to get a lot done during the day, start your day off with a nice, hot, fresh cup of evolutionist tears. Creationistclothing.com, right, brother?
Yep, exactly. Good one.
But how many times when we say, evolutionists believe that we evolved from ape-like ancestors, they say, you are an ape.
Yeah.
Maybe you're an ape.
Yeah, speak for yourself. I know your parents didn't do very good with you, so.
Well, going back to the classification schemes, let's go things that if you draw a circle big enough, Matt, are you not going to be able to basically throw anything into that circle? If we made a circle titled Things That Breathe, okay, human beings, you can see the picture of a baby, lions, platypus, gorilla. There you go, primate family, great ape, horses, things affected by gravity. We got humans, frogs, shoes. You got a bowling ball, a chair, motorized transportation. See, you can take modes of transportation. You can classify them in the same ways that we do creatures. Classify them based on their purpose, the number of wheels, the certain length from the ground, the type of vehicle it is, the transmission of it, the body of it, whatever. And you'll find that these modes of transportation are naturally falling into hierarchical patterns where you can have sedans more similar, obviously, to each other than to anything else, but then sedans will also be more similar to hatchbacks or even a van than they are to a tractor trailer. And so you can get these naturally producing hierarchies. I mean, it's not like human engineers are designing vehicles purposely to form them into hierarchical patterns. It just happens naturally. And in the biological world, when God created creatures, they naturally fell out into hierarchical schemes. I mean, I've heard some creationists say, hierarchies really is a good evidence for evolution. We got to answer. What's so good about it? This happens naturally, no matter what you're looking at. The way I've organized my books upstairs fall out into hierarchical patterns. That's why we say the differences make all the difference. And when we focus on these differences, I've done 100 formal debates, and I have in many of those debates brought up the Y chromosomal dissimilarity between humans and chimpanzees. And Erica tried to respond. Me and you did a full 2-hour response to her response about 2 years ago now. That's never been responded to. And so you'll see the human Y chromosome is entirely different to the pathetic chimpanzee Y. It's different in architecture, size, gene content. It's different in so many ways, but yet we are to believe that we share a most recent common ancestor with the chimpanzee, as in the chimpanzee is our closest cousin. We go back to a common ancestor here. So basically during this time, since the split, the chimpanzee Y had to change this drastically. And so notice your human Y here, brother, and your gorilla Y here. Notice how they're a lot more similar, even though humans and gorillas share a more distant ancestor than the human is to the chimp, the gorilla is to the chimp. That's within one family. Supposedly the great ape family, evolutionists, taxonomists put humans in the great ape family, and just within that one family essentially is a break in the hierarchy, and a break not in something minor, something as major as the Y chromosome that literally determines whether you're a male or a woman. At least the most rational thinking people. I guess these days maybe you could have a Y chromosome and apparently be a woman. But anyway, so there's a major break in the hierarchy. That's one thing we could look at, but that's one thing that's actually discriminatory. This is not agnostic to the debate where it's like, well, evolutionists can just look to sperm competition in chimpanzees and boom, problem solved. No, sorry, evolutionists, it's not that simple. Okay, they currently lack an explanation, and they even admit this. Notice here. So here's just a commentary article basically on a more comprehensive technical paper, okay? And they admit that their hypothesis needs to be evaluated in subsequent studies. And so whether it's the sperm competition or faster rates of gene conversion, whatever, it hasn't been tested. It's a hypothesis. And so sure, evolutionists, you can believe in your untested hypothesis, believe in your story to explain these massive discrepancies, but it's not science. And so take out of your textbook the claim that humans are undoubtedly related to chimpanzees, throw it in the garbage. It doesn't belong in a science textbook. Go ahead, Matt.
I like it. I don't know. It's just, this subject is just so vast. It's so crazy that we still have to do this. You know what I mean? It's like, it's unreal. I mean, you think the genetic bottleneck and the explanation of how mutations are accumulating based on generation times and how the hierarchical pattern that we actually see is the way we see it because of the generations that have already passed. Like, how come an orangutan looks older genetically than a gorilla? Well, their generation time is literally half. It's only 10 to 11 years old. How come the gorilla looks older than the human being and the chimpanzee? Well, because a gorilla's generation time is around 16 or 20 years, and then a chimpanzee is around 21 years, and then a human is about 30 years. So that pattern makes it look like there's a difference in ages when we all went through the bottleneck at exactly the same time. It's just unreal how simple things are in the biblical model and how convoluted it has to get in theirs. And they even invoke and made up a lot of their own things, like the mutation rate is based on evolution already being true. So they invented neutral theory for it. So everything mutates at exactly the same rate. It's like, that doesn't help you either. But they had to do it. They had to, because without it, evolution dies. It's why did they invent the phylogenetic mutation rate? Because the observable mutation rate destroys them.
I love it. Amen. And, that's why when these evolutionists, Matt, when we point to, from the technical literature, a mutation rate or levels of genetic diversity that is consistent with separate ancestry and young earth creation, or we look to function in endogenous retroviruses, pseudogenes, ALU sequences, the DDX11L2 gene, a lot of your evolutionists, they'll say, oh, but Matt, but Donnie, those authors are not concluding that separate ancestry is true. These authors are concluding that evolution is true. Of course they are. That's why they're invoking untested hypotheses, because their paper's not going to get published. Matt, if they were to conclude in the Y chromosomal dissimilarity paper, the major one that first came out in between 2006 and 2008, if they concluded, you know what, maybe humans aren't related to chimpanzees, do you think with that conclusion their paper would have been published?
You would have never heard about it. Ever. Matter of fact, I have a challenge right now for any evolutionist that might be listening tonight or tomorrow. I don't really care when it is. I want you to write up a scientific paper. I don't care what it's on. Just make sure like you know the science that you're writing and make it 100% factual, and then slap on a young earth creation title and submit it away. Watch what happens. It'll only take a couple days before it's rejected and returned to you. And then you can say, I guess I shouldn't be pestering them about peer reviewed, because it doesn't matter what it is. It's going to get rejected because it doesn't conform with what they want you to believe. How is that science? Science is about questioning, about going against the consensus, about raising the doubt. You think if anybody went against like big objects fall faster than small objects, where would we be if people were just like, oh, we just know the bigger ones fall faster. It doesn't matter. Who cares? Oh, the earth is flat. Who cares? Ignore all that nonsense. We don't have to worry about it, right? Well, we get stuck. It's people questioning the consensus that matters the most. It's always the people that are on the outside that do the most for science because they're questioning. And the other people are stopped questioning. Science is about questioning. So why do they hate us so much? They hate us because it's biblical. If we believed in a young earth and we weren't, we would be completely left alone.
Right. Amen. It's not about science at that point. The origins question to them, they go into writing these papers assuming that evolution is true, that humans are related to chimpanzees and the great apes, and they descend, to a past common ancestor. And so when they come across this data, guess what? Firstly, Matt, it's the data that we're concerned with. When we write our books and do our presentations and put out articles and do debates, we're looking at the data. For example, in my endogenous retrovirus handbook, paper after paper after paper, going over the many functional roles associated with ERVs, determining cell types, functional in embryological development, acting as antiviral protectors, functional in tumor suppression, performing viral mimicry, where they require the sequence similarity to exogenous retroviruses, okay? And so we look at the data, we look at the functional roles that we're discovering, and we conclude something different than what the authors conclude, because they invoke what? Oh, well, these must have been co-opted, with no evidence at all. The evolutionists want to blindly believe the conclusions, where we set the conclusions aside. We say, let's examine that. Is that legit? Is that scientific? Has that been tested? We're actually using what, Matt? Critical thinking skills.
Exactly. Yeah, exactly. And they have the audacity to say that we're stuck with what we believe in. If we were stuck, we wouldn't have become this way, because we were already evolutionists. We already believed that way, and we questioned it. We opened our broad spectrum in our minds and said, what's the reality of what's really going on? And the questioning led us away from it. They're not questioning anymore. They've swallowed it, hook, line, and sinker. They believe what they were indoctrinated and told to believe. And then there's creationists that are the same way. They were told, just believe this, and they don't know anything about it. So it's on both sides of the camp. Don't get me wrong. But the thing is, you're always supposed to question. How do you know if something's wrong if you've never studied it? That's the problem with Erica. She literally said, we're wrong about genetics. What was her reply about what she knew about genetics? Literally nothing. What in the world? So Dr. John Sanford, the inventor of the gene gun, who's published like over 70 papers in genetics, wrote Mendel's accountant, is wrong from the girl who's never studied genetics. I wonder why she said that. So critical thinking allows you to just go, oh, she just hates the topic. That's why she makes videos. That's why she makes fun of these things, and that's why she hates it so much. Why else would someone go so far out of their way? If the topic doesn't trigger them or offend them in some way, people just don't care. The second they care is when they actually show and they put their time into something like that. Of course they care. They can say they don't all they want, but actions speak louder than words.
Amen. Well said, brother. Truth defenders. So, Matt, man, basically your channel is in a way our secondary channel that I promote frequently, called Young Earth Creation. So if you go there, you're going to find a lot of material there. You're going to find a lot of re-premieres, novel content. if you go on our website, standfortruthministries.com, scroll down to the team, you'll see myself, obviously you, Matt. Matt and I, we've been doing this together for the last 4 or 5 years nonstop. So we found each other and we've just been having too much fun ever since, right, brother?
Yeah, man, we got together early on. I found him doing a debate. I said, "Oh man, this is perfect," because I wanted to work on editing. I wanted to learn how to edit videos, and you should have seen the first ones; they were god awful. But then what happened is I was like, "Hey, man, we should get together and do this together." I'm a new young Earth creationist myself, and we should get together. And I had this channel. It wasn't called Young Earth Creation at the time; it was a different name. But then I named it that. But it was just a backup channel. I literally had no real content on it. I just did random things. That's why if you go back in time, the videos had nothing to do with Young Earth Creation. And then I made the channel as a backup one, and we tried to grow Standing for Truth. And then I had a flood video go viral on my channel. So now my channel actually matters, because otherwise it was literally just trying to build Standing for Truth channel. That was the goal, is all of us, Team Standing for Truth, a group of Brandon and Sam and George and John. And we had a group of people. You can see right there, actually. There's the group of people right down below.
Yeah, right.
And yeah, that was like our team. So we formed this team, and then we would all do everything for the Standing for Truth channel, and then our other channels just they were just secondary. We didn't really care about them too much. But now we have content everywhere, because we started to flood YouTube with actual Young Earth creation content, fighting against the critics that the Young Earth creationists that exist outside of us weren't doing. Look at Aaron Raw. His videos were going unanswered, and he was literally criticizing every major organization. Now, Christians are supposed to turn the other cheek. That's great. I think that applies to something other than when they're ragging on what you believe in the scripture itself. So we came out with this new attitude where we would take what they would say, play their video, and then refute everything. We didn't do it in a mean way. I did it in a mocking way, honestly. I did a lot of laugh tracks.
Listen, did Elijah not mock the prophets of Baal?
Good point.
Nothing wrong with a little mocking, and unfortunately, well, put it this way, we're not into soft apologetics, I guess you could say. We do come in swinging, and you know what? The prophets of God did too. And these scoffers, like you said, that was the problem, especially with YouTube years ago when I was just watching YouTube, and like you, I wasn't involved. I noticed the creationists, they were just getting bamboozled by people like all these popular YouTube channels, Apologia, Vice Rhino, Aaron Raw, whatever, non sequitur show at the time. But there wasn't really any creationists countering it. And so when you'd look up Young Earth creation stuff, especially on YouTube, it was just all anti Young Earth creation stuff. And then, Ken Hovind, he started a YouTube channel, started doing content and taking on the evolutionists and debating again. And I thought, you know what? I got to get in on this. I got to get in on this war. And it's been 4 or 5 years now. And brother, like you said, we have content everywhere. And this has been a huge blessing, this channel, because now we can re-premiere everything. We've got content on every topic. We do videos directly address. See, unfortunately, and we know we get a lot of emails and messages, right, Matt?
Ooh.
Hey, have you seen this video from Gutsick Gibbon? Hey, have you responded to this video from Aaron Raw? And so unfortunately, some people who are possibly weaker in the faith, they might see a video from Dr. Dan Stern Cardinal titled Refuting Genetic Entropy, but then they don't find any counter to that video available. They might think Dan won, and they might fall away. They might lose faith. They might kind of shrivel up and think, oh, maybe there's not really a defense like I thought there was. But then, no, that's one of our missions, right? Is to basically take those videos, look at those critics, and refute them with sound, technical, and sophisticated arguments, and show the world, especially the world of Christianity, that, listen, we do have an answer to these challenges, like this one, the chromosome 2. So this is important. And possibly if we didn't come at Erica on this topic, she may have gone freely, making people believe that nope, there's nowhere else in the genome where you find the sequence in forward and in reverse.
Yep.
But as we usually do, we said this is probably another error. This is probably another false argument, false claim. We looked into it. God bless our brother Chris Roop, who ran the blast sequence and just completely decimated this challenge. And this isn't the first challenge we decimated. And so, you know what? Now there's material, there's content out there showing why arguments from Gutsick Gibbon are pretty weak. Go ahead, Matt.
Exactly. Like, I don't think people will realize how much content we released, and how many days of editing I would do. Remember, there was a time when I was releasing 2 videos a day with you.
Right.
I literally, you guys couldn't even imagine. One video took me 3 months one time, and I was only making videos, eating and sleeping. That was it. I mean, non-stop. We made a video called Refuting Aaron Raw's Flood Series, because his one flood video has 1.5 million views. So even if he made 1% of the audience listening to his content fall away, which I guarantee he did, because my best friend, Andrew, fell away because of Aaron Raw. He was a Christian, but he didn't know much. He just really was like, well, I guess if the Bible is true, like, these stories must be true. So he went to his pastor and was asking questions. He didn't get good answers. So he found out, he went to YouTube and he typed it in. And guess what? The answers came up. They came up with atheists. So Aaron Raw.
Hey, remember we were saying the other day, and this is true. I remember, and now we've done so much on it. I've written a book on it over here. I highly recommend Endogenous Retrovirus Handbook. Years ago, when evolutionists were using these arguments, endogenous retroviruses, chromosome 2 fusion, and so on, we would look on YouTube. You know, I wonder what the rebuttals are to these. There's basically nothing. On ERVs, there's maybe like a couple short little videos, kind of out of date, didn't really touch on much of the latest information on ERVs. And so now, we were joking around the other day, when you type in, you know, chromosome 2 fusion debunked or endogenous retroviruses debunked, basically all our material coming up on YouTube.
Yeah, that's it.
So the point is, the evolutionists no longer have they're basically not running YouTube anymore. They're kind of on the defense. Their arguments are now shown to be easily refuted, where in the past they had a little bit of a run of YouTube. There's a word I'm looking for, but I can't quite think of it because it's almost 6 a.m. and my brain's starting to shut down. But you know what I mean. You know, they're no longer paramount in the YouTube world, put it that way.
Yeah. I think we kind of exist for a lot of different reasons with our content. We supply the Christian with the answers that they're looking for if they want to defend the young earth creationist view. We're helpful to pastors, so if they want to take a literal view and a theological approach of young earth creation, they're going to need to know the answers of a scientific thing, or they can just recommend and say, go to them. I'm more theological, they're more scientific. We believe it's true based on faith. They have the evidence to back it up. So we want to be well-rounded. We really are for Christians. That's kind of the goal, is to equip everybody, but it's also for those that are on the outside that have only believed in evolution, who are like, oh, wow, I didn't know there was an alternative. Let's hear what they have to say.
Right.
And then all of a sudden we're like, wow, that Bible stuff is actually real? You mean that's a true history book? And we're like, it is. You won't believe it.
How many people, especially in our personal lives, friends, family, acquaintances, fellow coworkers, they have no idea about the evidence for separate ancestry, the evidence for a literal Adam, a literal Eve, the evidence in genetics for the Tower of Babel dispersal, the evidence in genetics for the flood bottleneck. All of this amazing evidence that comes together in a very consistent, sound, biblical model of origins. How many people have told us? I know people have told me in my personal life, especially in my family, I never heard any of this. You know, people in my family, like my parents, right? 50 to 60 years old. They've never been confronted with this data, but then they are, and they're blown away. They're like, wow, where was all this evidence my entire life? You know, where's it been hiding? And it's one of our goals, our ministry missions, to just pump the internet, pump YouTube, whatever, with endless content focusing on the very best lines of evidence for young earth creation, brother.
Yeah, and see, we didn't just stop there. We know that some people like watching debates, but we also know that some people like watching edited movies and videos. So I took that route. We also know that some people learn better from reading. So how many books do we have? 15? 20? I don't know. I didn't even count it. We have so many books. We've kind of covered all the bases. Like, we run a ministry basically by ourselves, and we've never even met.
Yeah, this is the closest we've come to meeting, which is why we need your support in the chat too. As we grow more and expand, we want to start implementing. We were talking about it probably a week ago. We'd love to implement some in-person SFT conferences where we can all come together for a weekend and just give some presentations, lectures, have some kind of conference conversations in front of people. I mean, that'd be a blast to do some in-person stuff, right? Rather than from our home offices all the time.
Yeah.
Or endless WhatsApp messages.
We'll probably have to go to one of Kent's Baptist boot camp things where he gets teachers to come, and like, you know, say like, let's all meet there, and then we can just randomly show up there and give lectures.
Right. Let's all meet at Dow for a standing for truth and creation science evangelism fusion boot camp, which I think would be a blast. I think the reason why Erica did not find any of these repeats and forwards and backwards in the genome is because of this, Matt. Tell me if you agree, brother.
Genetic entropy is something that he mentions a lot in this book. Genetic entropy is really, really stupid.
But I'm also a geneticist and that's not really my world. Yeah, I'll be the first one to admit that I'm not a geneticist.
Again, I'm not a geneticist. I can't say for certain genetics is not my forefront.
Because I'm not a geneticist.
Again, not a geneticist, and I'm not a geneticist. I'm not a biochemist.
I'm not a geneticist.
We should have a conversation about genetic entropy. I've heard you speak to like CRISPR and stuff like that before, and I would be very interested to have that chat. I'm not very, well-versed, as I said, for like the 20th time. I'm not very well-versed in genetics.
Yes, so here's the thing, and I'm going to use this to segue into genetic entropy because I feel like that's kind of where you're, pulling a little bit, which is fine because I got my notes right here because I don't know enough about genetics.
Again, I cannot emphasize enough how this is not my field. I would, as I continued to kind of begrudgingly admit throughout the course of this conversation, that genetics really isn't kind of my field. I can talk to you a lot about, other stuff, but genetics isn't it. I don't know about because this isn't my field. Genetics really isn't kind of my field.
We're not going to be touching very much on genetic entropy. I feel very comfortable hand waving it.
That's not my field. I wish I could give you a better answer. I can't give you great answers because I don't know them.
Oh, idiot.
When we see genetic entropy, I'm going to say genetic entropy, and I'm going to do the genetic entropy sign, and we're going to say it's like this, and then I'm going to say skip. So I'm going to be like genetic.
Oh yeah, I remember making that one.
One day we need to go through an SFT classics night and just go through all of the old school videos that you spent all night making, but we kind of spent the entire night together brainstorming. Like, hey, how amazing would it be to get all of these moments where Erica was cornered in a debate? Let me say a couple things. Erica's videos, she's getting more and more snarky, I've noticed. So she throws a lot of jabs, but we got thick skin. I don't care. Most of the time I find it funny. And so she can handle that. I think it's fine. She doesn't mind. Yeah, but secondly, the critics will say, oh, but it's so noble. It's so noble of the evolutionists when they're backed in a corner in a debate on an argument. And then they say, oh, I'm not a geneticist or I'm not a geologist. But that's not the point. Matt, obviously people can't know everything about every subject. The fact is, and why this is so relevant, is because Gutsick Gibbon, correct me if I'm wrong, Matt, makes video after video after video, or when she debates lesser informed creationists, what arguments does she use? ERVs, chromosome 2 fusion, genetics related arguments. Or she hand waves away genetic entropy, says it's not a real thing, says it doesn't exist, says it's anti-science. Then why every time you're challenged with a point on one of these topics, you say you're not a geneticist? That's why it's relevant. That's why it's important. It doesn't have to, it's not about, oh, she's saying she's not a, yeah, we get it. Not everybody can be an expert at every topic. But if I was doing video after video after video on astronomy, saying the Big Bang is completely whack and anti-science, but then when I'm debating the topic, I don't really have an answer to anything on it. I mean, you know what I'm getting at. It doesn't look too good on me.
Exactly. Their goal in a debate is to find what you don't know and then ask you a question about it to trip you up. Because they, like you said, you can't know everything. So their goal is to find what you don't know and make you say, I don't know. And then they claim victory. Yet when they say they don't know, they're claiming like this is the ultra thing that we should possibly be doing. Remember when Erica went to the Ark Encounter and she was like, she walked up to like Jason Lyle, who's an astrophysicist, and asked him something about like the Noah's Ark and the flood layer. He was like, you probably want to talk to our geneticists over there. And then she was like, oh, they don't even know about their, it's like, well, wait, you're claiming yourself that that's a bad argument. Now you're using it. Just unreal.
Well, let me respond to this comment. James Baker. He says, okay, Donnie, people love spelling. Like 99% of the time my name is spelled in a comment section or a live chat. It's IE, right? Gutsick Gibbon's latest video. Everybody's just talking trash about me, right? And I don't care. I get a kick out of it. You know, my power level increases every time a critic scoffs at me, basically. So anyways, DONNY.
It's on the screen. No, I can see it.
No, but it's 99% of the case. I get a kick out of it. Anyway, so James Baker, appreciate the comment. He says, okay, Donnie, you caught her, maybe. But your boast is that she wasn't correct about something. Okay, it's not just this one thing. How does that debunk human evolution? He says this discovery is all we have. Okay, chromosome 2 fusion. Let's quickly, in a nutshell, in a minute. Take a deep breath. Okay. So the so-called fusion sites, 2 degenerates, corrupt. It's only 798 base pairs. And guess what? It's overlapped by a highly functional and essential gene, the DDX11L2 gene. The critics like Erica say, oh, it's just a pseudo gene. The activity there, it's spurious. It's noise. We've demolished that tonight in the article series. Anyways, the so-called centromere, cryptic centromere, the centromeric DNA, the alphoid repeats. Guess what? Everything there, that so-called signature, overlapped by, yet again, a highly functional and essential gene. So basically what we're looking at in terms of these so-called signatures are actually functional components or functional elements of highly functional and essential genes. Those telomeric repeats, they're found all throughout the genome, and they function in gene expression. They're necessary for the health of the cell. And we find instances of those sequences in both forwards and reverse. That's it. So it's not just that one thing. It's not like, oh, that's all you have. No, every aspect of the fusion argument has been contested. And would you agree, Matt, have been overturned?
Well, he's saying, how does that refute evolution? From the evolutionist's mouth himself, Kenneth Miller. He said it, not us. So take it up with him. He's the one that said, if there is no fusion, there is no human evolution. There is no common ancestor. Well, there it is. So I guess you could take it up with him. Or that's my best.
Dr. Ken Miller, I mean, who's he? He's just one of the most prominent, famous evolutionists who I'm pretty sure had an active role in the trial there. What was the name of it?
Trial? Yeah, the Dover trial.
Yeah. And he presented, as far as I know, the fusion.
Yeah.
That was his main reason to say that it's science and should be taught and all that.
Yeah.
So yeah, he said, you know what? If we don't find it, evolution is wrong. We don't. I mean, that's pretty. He's pretty clear. I mean, he's pretty adamant there. That's pretty straightforward. There's no other way to interpret that.
Yeah. He put themselves out on the ledge too. He put all his cards on the table and then put everything on the edge of the cliff. So that's how confident they are and how much they need it.
Imagine putting all of your cards, all of your hope, all of your dreams. Their dream is to be related to a banana plant. Amen. What a dream. What is your dream? To have a dream.
Yeah.
So, right. And so all of their hopes and dreams, all of their cards on one argument, the chromosome. Now, Ken Miller, nothing else matters. Homology, ERVs, nested hierarchies, pseudogenes. None of it matters, Ken, because he admitted here, no fusion, no common ancestry.
Case closed. Yep.
That's it. It's over. It's done.
Yeah.
Well, Matt, this has been fun. We got to do a part 2 soon. We're going on 3 hours. What do you say? Ooh. Should we start winding things down?
Yeah. I know. It's late. We got to go. That was way too long.
That was way too long, way too fun. You could tell as time went on, we got a little bit more animated, laughed a little bit more because the later it got, and we might be in overtired mode. So we better wrap it up. But one could say this was a thorough demolition of the chromosome 2 fusion. We had some fun interaction with these clips. And so leave a comment if you liked this show. It's impromptu. We kept it organic. I surprised Matt with a little clip there from Erica. We had some good feedback on it. We didn't really get much into the comments on comments, Matt, but maybe we'll do that next time. Yeah. And if you enjoyed this kind of format, the way we ran this show, let us know. And if there's any videos you'd like us to respond to, comment on like we did tonight, send it to us, standingfortruthministries@gmail.com, and we'll get to it. So, Matt, any last words, brother?
Yeah. Check this out, everybody. Oh. Shroud of Turin replica coming out.
You are the only man on the planet with that. That is one of a kind right there, brother.
For sure. Life size, too. I'm going to get it nice and strong and sturdy so I can take it around and give talks about it as well.
Did you have to travel to Rome and infiltrate the Vatican in order to get that? That's what you've been doing the last several months, eh? National Treasure starring Nicolas Cage type stuff.
Ran away with it. They'll never find it again. I replaced it with something that's ridiculous. Yeah.
Right. Yeah. Like National Treasure. Didn't he go in and sneak in and replace the Declaration of Independence or something?
Yeah.
you did it, brother. You did it. Christian Pike is asking a question about horse evolution. That's a good question. And you know what? We'll save that for a week. That'll keep us going for a while. We'll save that for a full video in the future. So anyways, 3 hours, been fun. Share around this content all. God bless. Hope you had some fun, and you'll see us soon. So Matt, peace out, brother. Stand for Truth is out.